Two. Oh.
Continue with the workshop on genotoxicity that we are starting a little bit late, so our apologies for that. But Dr. Laudermende is going to facilitate this workshop for us, and I'm supposed to introduce you first. That's what comes up on my script. So our introduction of the workshop organizer.
Thank you.
So hold on just 1 second. I think you're up next. That's what the script says. Dr. Laudermende obtained his PhD from the National University of La Plata in Argentina in 1978. He was appointed as a member of the research career of the National Scientific and Technological Council of Scientific and Technical Research of Argentina in 1981, and is today a senior researcher. Since 1991, he serves as professor of molecular cell biology at UNLP. He is the author of more than 450 contributions in the field, including scientific publications mostly in high-impact and prestigious scientific journals, research communications, and conferences worldwide. He's a regular lecturer at the International A. Hollander courses organized by the International Association of the Environmental Society. He's carried out postdoctoral research activities at the National Cancer Institute in the United States, and has been a guest scientist at the University of Helsinki in Finland. He is an expert in molecular cytogenetics, genetic toxicology, and ecotoxicology. He is or has been a referee for more than 20 international scientific journals, including some of the top periodicals within his work area. He's the editor of 8 books in his field and currently is the head of the Laboratory of Molecular Cytogenetics and Genotoxicology at UNLP. So I believe it is the floor is open for you to introduce the speakers in the workshop.
Yes, unfortunately, the only data I have from each of the speakers is the name and the affiliation.
Let's go with that.
Okay. Do you
Sounds
think so?
great. I think that sounds great. We'll go ahead and get you started, get your presentation set up.
Okay, first of all, there was a mistake in the program. The title of the workshop is missing. The title is something like From Laboratory Studies to Marketing Regulation. And the first speaker is Dr. Leon Stakowski. He is PhD, and he is consultant in genetic toxicology. So
I'll start from there. So again, Leon Stankowski, been doing gene tox for around the is either you can use E. coli tester strains, WP2 UVRA, or that same strain with a plasmid inserted, PKM101, or TA102. And they detect base pair substitution mutations, largely at AT pairs. These are all engineered strains, such that they're typically deficient in DNA excision repair and/or error-prone, they have an error-prone DNA repair function, which makes them more likely to revert back in the presence of a mutagen. And the E. coli, or the salmonella strains have a leaky cell membrane so that large bulky molecules can get in more easily. Typical assay design is you'll treat 10 to the 8th bacteria per plate in the presence of limited histidine or tryptophan. It allows all the cells there, even though they're histidine or tryptophan auxotrophs, to go through a couple of divisions and form a typical background lawn. Normal study design would be usually 5 or more dose levels, and in this situation we want to test up to 5,000 micrograms per plate, which is the limit dose, or solubility or toxicity limit, which is determined by looking at the background lawn. All those bacteria, the 10 to the 8th bacteria, have a chance to go through a couple divisions. You get a hazy, cloudy background on the plate, but in the presence of increasing toxicity, that lawn thins out and starts to clear. Each study should have positive controls, which are diagnostic for each of the tester strains. You want your frame shift tester strains to pick up frame shift mutagens, and likewise base pair substitution frame strains to pick up base pair substitution mutagens. We'll also include a vehicle control for all the strains, and all these studies are done in the presence and absence of S9. S9 is essentially a rat liver milkshake, which is an exogenous metabolic activation system. It contains liver homogenate from air chlor-induced rats or other similarly induced rats with the appropriate cofactors and energy supply. Typical experimental design is all done in triplicate, incubate the plates for 48 hours, and you'll count them. And some representative plates are shown here. Typical positive response is the requirement for a 2- or 3-fold dose-dependent increase depending upon the strain. The strains with the lower background, spontaneous backgrounds, typically require a 3-fold increase, but the higher background strains where the revertant frequency might be typically 100 per plate would require only a 2-fold increase. So I'm going to shift now to mammalian cells. There are 2 main assays that are performed these days: the mouse lymphoma assay, MLA, which looks at the thymidine kinase locus, and the CHO HPRT, or maybe the V79 cell line HPRT assay. The difference between them involves the arrangement of the target genes within the cells. The mouse lymphoma, the target gene is heterozygous. One of the copies is knocked out. So you can get a gene mutation that only involves the TK gene, resulting in a point mutation or small deletion, or you can pick up much larger damage because there's that second chromosome there that has the originally inactivated TK allele. That imparts essential sequences or functions up and downstream from the target gene. So if you knock out the target gene and extend into those adjacent essential sequences, there's another copy present to provide that function, and the cell can survive. In contrast, the HPRT gene is hemizygous. It's located on the X chromosome, and although you can detect gene mutation very easily, it seems that large deletions that extend into those other essential adjacent sequences are lethal. So those mutations don't yield viable mutant colonies. The guidelines in questionnaire 490, as I mentioned, covers the mouse lymphoma assay and the TK6. Again, the target gene is autosomal, and you're going from a TK plus minus to a TK minus. Those cells become resistant to trifluorothymidine, which is a metabolic poison. 476 covers the CHO HPRT assay and the less commonly used XPRT variant. Their HPRT is X-linked, and you go from a hemizygous to a HPRT minus. Those cells are resistant to thioguanine, a metabolic poison. So those are the basis, the chemicals that we use to select mutant versus wild type cells: trifluorothymidine and thioguanine. So both cases, you typically want to treat enough cells to have 2 to 3 million survivors per culture. Normal study design is about 4 dose levels. Again, typically we would use more, especially if there's going to be expected toxicity, so that we can have 1 or 2 to throw away if need be. Limit dose in this case for OECD compliance is 2,000 microgram per mil or 10 millimolar, or the limit of solubility or toxicity. In this case, it's 10 to 20% relative survival or relative total growth, depending upon the assay. Again, positive and negative controls are required. test in the presence and absence of S9, and experiments are typically done in duplicate, although single cultures would be allowed, but they would recommend that you increase the number of cultures or dose levels so that you typically have about 8 cultures to analyze regardless. After the treatment, we typically subculture for 2 to 7 days, depending upon the test system. We select mutants, as I mentioned, with trifluorothymidine or thioguanine. We do concurrent cloning efficiency so that we know what the viability of the mutant selection population is. Incubate those for 7 to 14 days to allow colonies to develop, and then we count them. And there are representative examples there of the CHO-HPRT assay on the upper right. The bottom left is soft auger cloning for the mouse lymphoma assay. There's also variation where you could do fluctuation analysis to determine mutant frequency for mouse lymphoma assay, and those are the 96-well plates you see on the bottom right. Again, a significant dose-dependent increase is required for a positive response. This one has an additional requirement to either exceed the 95% control limit for your negative historical control values or the global evaluation factor in the case of the mouse lymphoma assay. And that's a empirically determined number that was arrived at based on a number of international workshops and collaborative data analyses. The other main type of assay, again, mammalian cells, are cytogenetics assays. And we can look at chromosome aberrations under the 473 guideline or in vitro micronuclei under the 487 guideline, which I'll talk about a little bit later. The assays can be done in human peripheral blood lymphocytes. There are modifications where you could look at primary rat peripheral blood lymphocytes. Also CHO or CHL cells, for example, are commonly used. And again, we're looking for either structural aberrations, changes, breaks, or rearrangements induced by clastogens, or we could also look at numerical aberrations, for example, polyploidy or endoreduplication induced by antigens. These particular types of assays, the 473, are not designed to pick up plus minus 1 or 2 changes in chromosome number, because you can run into that just randomly when you make your spreads. For example, if you're doing a CHO assay, chromabs, you've got typically about 20 chromosomes, and you may score anything that has anywhere between 18 and 22, thinking that you might get random loss during slide prep. Assays can be done typically in either centrifuge tubes for suspension cultures or in flasks for monolayer cultures. You want to treat exponentially growing cells because you need cell division to fix and allow manifestation of the chromosome aberrations. So primary cultures are stimulated with mitogen, for example, PHA. Typically want to have 3 scoreable dose levels. Again, the same limit dose as in the mammalian cell gene mutation assays. Now, though, we're looking at a toxicity limit of 40 to 50% relative population doubling or relative increase in cell counts or mitotic index in the case of human lymphocytes or rat lymphocytes. Same kind of considerations, positive and negative controls. In this case, we have a short treatment with and without S9 and an extended treatment minus S9 only. And they can be done in either single or duplicate cultures. However, the requirements for scoring are the same. So you want to harvest all your cultures, regardless of the length of treatment, roughly 1.5 cell cycles after the start. And the cell cycle time obviously is cell type dependent, so it varies. You want to arrest the cultures in metaphase with colcimid or colchicine so that you accumulate the metaphase spreads or metaphase cells. And you want to score 300 per dose level. So if you got duplicate cultures, you could do 150 per culture. If you do single cultures, 300 cells still per dose level. Same old story, looking for a significant dose-dependent increase. And again, you want to exceed the 95% control limit for a bona fide positive response. And some examples here from CHO metaphase spreads are for exchange aberrations or breaks there. You can see polyploidy on the right-hand side metaphase spread. Here's some examples of human lymphocyte metaphase spreads. You can see a dicentric chromosome on the left and a break on the metaphase spread on the right. And as you might imagine, looking at these spreads, which can sometimes be a lot like spaghetti or rigatoni maybe, it takes a pretty skilled technician to go through and score them in any reasonable fashion, any reasonable speed. So one of the things that was developed over the course of the years is an in vitro micronucleus assay. And it's uses the same kind of test systems. Also, TK6 is pretty common here, looking for structural and numerical aberrations using the same kind of test systems. But in this case, we can differentiate between things that cause chromosome breaks and things that cause chromosome loss, clastogens versus antigens. And the assay is typically done in two different manners. One in the presence of cytoclasmin, one in the absence of cytoclasmin B, and that can be used for any kind of established cell line where you're looking at population doubling type parameters for cytotoxicity, where you're looking at human lymphocytes or peripheral blood rat lymphocytes, where you don't necessarily know you're stimulating every single cell there. You don't have a good handle on which ones are undergoing division and which ones aren't. You are required to use cytoclasmin B. And the difference is, without cyto B, you form two daughter cells. Regardless of the mechanism, clastogen or antigen, you should show up as a micronucleus in one of the daughter cells. If we use cytoclasmin B, it inhibits the cytokinesis step of mitosis and cell division. So you end up with what are called binucleate cells, typically, again, with the presence or absence of a micronucleus if an event, clastogenic or antigenic event has occurred. And as I mentioned, for lymphocyte cultures where you don't know that all the cells are dividing, you want to use cytoclasmin B so that you're looking specifically at scoring binucleated cells, making sure that the cells have gone through at least one division. Assay design is very similar. Again, treat exponentially growing cells. You want 3+ dose levels. We're looking for 50 to 60% decrease in relative population doubling or increase in cell culture or cell counts in cell lines, or a parameter called CPBI, or replicative index in primary cells when you're using cyto B. Again, positive vehicle controls across the board, short treatment with and without S9, extended treatment minus S9. You can use singular duplicate cultures. In this case, you're going to harvest at 1.5 to 2 cell cycles after the start of treatment. And we are going to score 2,000 cells per dose level, whether it's singular duplicate cultures, 2,000 per dose level. Same kind of parameters, looking at significant dose-dependent increases that exceed the 95% control limit for your laboratory. And you see examples of micronuclei, heme-sustained cell on the left, a binucleated cell, and a binucleated acridine orange stained cell on the right. One of the beauties of the in vitro micronucleus assay, though, is that you can differentiate mechanism. So the chromosomes have centromeric sequences and kinetic cores that are required for proper cell division. And depending upon the presence or absence of those kinetic cores or centromeric sequences in the micronucleus, you can determine whether it was responsible, whether a clastogenic event occurred and only a fragment of a chromosome was there, or whether it was an antigenic event and a whole chromosome was present. So in cartoon form, you can look at here on this slide, you can look at mechanism of action if you stain with either pan-centromeric DNA sequences that detect the centromere, or if you use immunologic methods, CREST antibodies, and you can detect the kinetic cores. So the cell on the left is a kinetic core or centromere negative micronucleus, which means it arose by a clastogenic mechanism. On the right, that micronucleus has either a kinetic core or a centromeric sequences, and you can tell that it's a whole chromosome and it arose by an antigenic mechanism. Here are some actual slides pulled off, stolen from the internet. You can see metaphase cell on the top. It's a pan-centromeric DNA stain. You can use that for mice or rats because there are probes available that cover every single chromosome in those karyotypes. And you can differentiate the C+ on the top and the middle, centromeric sequence containing micronucleus, and differentiate that from the C- micronucleus that has no centromeric sequences on the right-hand side. Likewise, in the bottom panels, it's CREST staining, propydium iodide, stain on the left, the crest staining in the middle, and when you overlay the two, you can differentiate which micronuclei contains crest antibody binding kinetochores and which ones don't. And I think that's about it. I could take some questions if you have them.
We have time for at least a couple of questions.
I was just going to ask one quick question about the S9 treatment. So that's animal-derived
Yes.
enzymes. But has anybody looked into what would be a more human relevant mix of enzymes to do?
So anybody that typically gets a positive response with S9 only, the first thing they want to think of is what happens with human S9. Obviously, human S9s, except for a few individuals who maybe are breaking the law, don't induce their liver enzymes to the same extent that we can in animals. Any comparisons that have been done, a positive response is typically much lower or it disappears in the presence of human S9 compared to rat S9. But the go-to standard for regulatory purposes is erichlor-induced or phenobarb beta-naphtoflavone-induced rat liver S9. Where you could make a case for human versus the rat and it making a relevant difference is where in vitro or other data, metabolism data, shows that particular metabolite that's causing the problem with the rat S9 doesn't even occur in the presence of human S9. But without that kind of additional information, you're basically going to be talking to the wall as far as, with the regulatory agency.
So there's going to be too many differences
From individual to individual, and in fact, what they typically do is they'll pool donors. So you get maybe a little bit more representative, more uniform response, but even then, it's so different, lot to lot, batch to batch, person to person.
Leon, what's your suggestion of using some sort of metabolic active cell lines like the HIP-G2 instead of using a conventional CHO B79 plus or without S9?
There are some metabolically competent cells, and they're starting to work their way into the guidelines that you can use them. But the validation work hasn't been all that robust. If I can imagine like a big pharma company that wants to do that. They would have to, basically go through a whole validation exercise to look at a whole host of known S9 requiring mutagens to show that their test system would work. I think the agency would buy it. The agencies are typically about good science, but you got to cover all the basics first and then add on to that.
Okay.
And if anybody thinks of any other questions, effective next Monday, you can reach me at will.com, willresearch.com.
Or we can have time some plain discussion after the workshop.
Okay.
I think it would be a wise idea.
Correct. I think other questions will arise as they hear more talks and more systems.
Right.
Okay.
Okay, let's move to the second speaker, who is Dr. Rohan Kulkarni. He's Director of Genetic Toxicology in BioReliance in Rock Hill, Maryland. Do you need some help? No.
Good morning, everyone. My name is Rohan Kulkarni, and I'm the Director of Gene Tox at BioReliance. I'm going to talk about today the basic aspects and most commonly employed in vivo assays. And I'm going to take some cues from Leon's talk because some of the things are common in both the assays. So before I go into the in vivo assays, I want to give you a more of a historical perspective of how the in vivo assay or how the gene tox assay started. So it all started from, in 16th century, there used to be the epidemiology assays. And the problem with these assays is there are a lot of ethical issues because you had human volunteers. Also, there was a long latency and high background. And also, because of these issues, it made these assays more impractical. So in 1915, the rodent bioassays were developed, and it used to take around 5 years, and it used to cost $4 to $5 million. In 1970s, this is an important era for gene tox, and in this time, in vivo gene tox assays and the in vitro gene tox assays were developed. And the reason we study gene tox, because as per the James and Elizabeth Miller, there's a quote by them that many carcinogens are mutagens and the most mutagens are carcinogens. So if you know if your compound is a mutagen or not, you can predict if it is a carcinogen or not. So you want to know if your compound is going to cause carcinogenicity. So the in vivo assays were developed in 1970s. Again in 1970, as I mentioned, Bruce Ames discovered the Ames assay, and those assays are major workhorse for the gene tox assays in today's world. In 1990, screening assays were developed, and these assays basically, they are the fast turnaround time and they require small amount of test article, and my colleague Kamala is going to talk about that in her next talk. And in now 1990s, the new thing is most of the companies are looking for the SAR or the QSAR related test. So these are the in silico assays where you do computer-based analysis to see if your compound is genotoxic or not. So today's topic of discussion, I'm going to talk about the most commonly assays, which is in vivo micronucleus assay, and I'm also talk about the in vivo mammalian alkaline comet assay. I'm also going to touch upon 2 or 3 less commonly used assays, one of which is the TGR or the transgenic rodent somatic and germ cell mutation assay, which is also known as the Big Blue assay, and the in vivo chromosome aberration assay, and the latest kid on the block, PIGA, or the in vivo gene mutation assay. So again, about the in vivo micronucleus assay, as I mentioned, this assay was developed in 1973. John Heddle developed this assay, and the assay is basically to assess the potential for DNA damage, which might alter the chromosome structure, or it could interfere with the mitotic apparatus. So this assay could detect the direct DNA acting agents or the non-direct DNA acting agents. So in vivo micronucleus, it detects micronuclei, and it could, with the micronuclei, you can look at the clastogenicity or the anagenicity of the compound. Also, this serves as biomarkers for cytogenetic damage. However, this particular test has a major requirement, which is exposure. So when you do this test, you have to show that the animal is getting exposed to the compound you are administrating. If there is no exposure, there is no test. And I'm going to talk more about this in my next slides. So this is the theory behind how micronuclei assay, in vivo micronucleus assay is done, or the science behind it. So animal is injected with the test compound. The stem cells or erythroblast, after the final mitosis, goes into the polychromatic erythrocytes. It develops PCEs. Now the top portion of it is how the normally what happens. So it becomes the PCE. In the maturation stage, the nucleus is expelled, and you have a normochromatic erythrocyte or NCs, whereas there is no nucleus present. However, if there is any chromosome damage, you have, the main nucleus get expelled. However, the micronucleated, the micronucleus or the small portion of the chromosome remains in the nucleus, and that creates the micronucleated NC. So what are the exposure methods? How this test is conducted? As Leon mentioned, there are OECD guidelines to guide how to conduct this test. And this can be done in rat or in mouse or any suitable mammalian species. However, most of the assays are done in only rat or mouse, and there is no preference which animal model you will use. It depends upon your compound and what data you have from your previous studies. When you get the animals from the suppliers, the weight variation should be within 20% of per sex. Now, I mentioned that if there is no exposure, there is no test. So what are the different ways to administer doses? So you can do oral gavage, you can do IP, intraperitoneal dosing, intravenous dosing, subcutaneous or dermal dosing. Now, if you are going to do intravenous dosing, you don't have to show exposure separately, because intravenous will administer drug directly into the vein and it goes into the plasma. And we are going to look into the bone marrow for the endpoint. And bone marrow being the highly perfused organ, when it goes to plasma, it is understood or it is implied that it will reach bone marrow. However, for oral gavage or the subcutaneous or dermal application, you have to show that there is exposure. Now, there are a couple of ways you can show exposure, which I'm going to talk in my next slide. The intraperitoneal route of administration, although it is a valid route of administration, as per the FDA guidelines, it is not a desired route of administration because it's not of human relevance. For dose formulations, you can use solids or you can use liquids, and you have to freshly prepare the dose formulations unless you have a stability to demonstrate that at different temperatures. How do you select dose for these studies? So here are some terms. You have to select the maximum tolerated dose. And what does that mean is when you administer dose to the animals, there should be some clinical signs because of this dose, and it should not cause mortality. If you have mortality, that means it is not a tolerated dose, and you have to go for a lower dose. I told you that there are different ways to show exposure. Another way of showing exposure is to see the bone marrow suppression. So if you know your compound is going to cause bone marrow suppression, you can look into the reduction of PCEs to NCE ratio or inhibition of erythropoiesis. And that shows that your test compound has reached bone marrow and it is decreasing or it is inhibiting the erythropoiesis. Also, you have to select a dose that does not disturb animal physiology. And you have to use the highest dose for the definitive study. Otherwise, you can use the limit dose. So for the in vivo micronucleus study, the limit dose as per the guidelines is 2000 milligrams per kilograms per day if your study is going to be less than 4 days, less than or equal to 4 days. If your study is going to be more than 14 days, you have to, the dose drops down to 1000 milligrams per kilogram per day. There is another concept called as maximum feasible dose. Now, I told you that you have to prepare the dose formulation from the solid or from the liquid. Now, in case of solid, if your vehicle, what you're going to use, if it is not going to formulate the highest amount required to reach the maximum tolerated dose, you can justify saying that this is the maximum feasible dose we can prepare based on the dose volume and the solubility, and we cannot go more than that, which is acceptable, but you have to show that this is the top dose you can reach. Also, in gene tox studies, safety multiples are not appropriate. Now, what is the meaning of that? The requirement for the study is you have to go for 2000 milligrams per kilogram per day. But you cannot justify to the agency, whether it could be FDA or any European agency, saying that the human exposure is going to be only 1 microgram. Why do I need to go to 2000 milligram per kilogram per day? This is not a risk assessment. This is the hazard assessment. That's why you have to reach the required dose as per the guidelines. Now, this is the typical study design for a definitive assay. So once you select the MTD or MFD, the first step you do is you formulate the dose. As I mentioned, on the same day, or if there is stability, on the previous day or before that. You do the dose administration by the method which we already discussed. You can also do the TK samples. For example, if you know the compound is not going to cause any bone marrow suppression, and if it is an oral gavage, you have to show that the compound is reaching plasma. So in that case, you will collect the TK samples or the blood samples, and you will show that there is exposure in your plasma. You also look at the clinical observations, and this is the important portion of these studies, that you have to show that the animals are getting the drug, and there are some clinical observations and not mortality. Now, there are a couple of study designs. You can either do the 1-day dose or the 2-day dose. If you are doing the single dose study, you have to collect bone marrow 2 times, 24 and 48 hours after the last dose or the dose administration. If you are going to collect the chromosome aberration, if you are going to do the aberration studies instead of micronucleus, you have to treat the animals with culture meat prior to sacrifice. And once you sacrifice the animals, collect the bone marrow cells, you treat the cells in the hypotonic treatment fix the cells, and put it on slide. Now there are two ways you can analyze the cells. The one traditional method is a slide-based micronucleus evaluation, or the new method is the flow cytometer-based method. And definitely flow cytometer-based method has advantages. The main advantage is there is no human bias, because these cells, the bone marrow cells on slides, are scored by human scorer, and there is a chance of error doing that. However, for the flow-based method, it's automated system, so there are less chances of error. Also, there's a limitation of how many cells can be scored by human scorer, and as per the new OECD guidelines, you have to score 4,000 cells. 4,000 cells per animal in a group, and in group it becomes 20,000 cells. However, with micronuclei, since it is an automated and machine-based system, per animal you can score 20,000 cells, and that increases your statistical power. So we have seen that many clients are going for the flow-based analysis if it is possible. And this is a few photos I want to show how micronuclei looks. So this is the PCE, and this is the micronucleus. And these are the PCEs without the micronucleus. So in summary, the guideline requirements are you have to have a 2,000 milligrams per kilogram limit dose, or the MTD, or the MFD, maximum tolerated dose, or the maximum feasible dose. Bone marrow exposure should be achieved in a single or multiple treatments. Now, the advantage of method is it is possible to show that the bone marrow exposure by the bone marrow suppression or the PCE to EC ratio. You can also collect the blood samples, the TK samples or BioA samples, to show the plasma exposure. And the in vivo system takes benefit of the intact metabolic processes. So that's the main benefit against using in vitro systems. Because in vitro system, even if it is close to S9, for example, S9 fraction is used to mimic the liver enzymes. It is not a complete system, and that's why the in vivo systems are superior in that way that it shows the entire dynamics of ADME. In fact, we have seen that if your compound comes positive in vitro assay sometimes, the positive effect can go away in the in vivo assay, and that's because there are DNA repair mechanisms in the animals which could take care of those compounds as well. And as I mentioned, the main disadvantage is without exposure, the test is not valid. So if you fail to show that the compound is not reaching bone marrow, it is an invalid test. The second test assay I want to go for is the COMET assay. We heard a lot about COMET assay this morning, and this is also known as the single cell gel electrophoresis or COMET assay, and this detects the DNA damage and repair individual cells. This can be done in vitro and in vivo both, and this is done under alkaline conditions where pH is more than 13. It can detect single or double strand breaks. Single strand breaks as a result of the alkyl labile sites, and it can also detect the nucleotide exchange repair. This is the typical or the representative graph of how the COMET assay or the COMET slide looks. So this is the nucleus, which is in the center, and the DNA damage is correlated to the length and amount of DNA fragments which are migrated outside the cell nucleus. So more the tail you have or more the fragments you have, that shows that there is a more damage. This is the history of in vivo validation for the in vivo COMET assay. So this started in 2006. In the first trial, there were 5 lead labs, and BioReliance was one of the lead labs, and Kamala Pant was one of the main person on the who did the assay validation, and she was also part of the OECD guideline committee. So in 2006, the first assay was done with 5 labs, and only 1 positive compound was selected. In the second stage, which was in 2007, 5 labs did with EMS as a positive control, and they used the 3 coded chemicals to see that how the coded chemical also is there getting a similar response or not. Based on the results, on the third trial, the protocol optimization was done to increase the the within lab reproducibility. Then in the fourth step, the lab recruitments were done. There were more labs. There are 13 labs which participated in the validation to confirm the within and lab reproducibility. And in the fourth stage, which is the predictive capability, 14 labs were involved and there are 40 chemicals which were done. So as you can see, it started in 2006 and the validation went up to 2011 and beyond. Then an intent was filed with OECD to develop guidelines for this. And in 2014, September, Comet assay has guidelines now. And so this is an OECD accepted assay. So the question comes is when to perform in vivo Comet assay. So as I said, this is an in vivo assay. So along with the in vivo micronucleus assay, this can also be used as a second in vivo test. And this can, the beauty about this assay is this can do in combination with the in vivo micronucleus assay, which makes it as a combination assay. And the main benefit of doing the combination assay is, you save animals because you use the same animal for both the endpoints. You can look at the Comet assay and the micronucleus as well. And this can further evaluate in vitro positive finding. So if you have any in vitro data which is positive, you can go into in vivo assays. You can look at the micronuclei, you can look at the Comet assay and see if the data is reproducible or not. If the positive response what we saw in vitro, is it going to see in vivo as well? You can also look at the tissue specific gene tox activity. For example, if you know your compound could be metabolized in liver, you can specifically look into liver to see if there is a DNA damage. If you know your compound is, the first set of contact is going to be esophagus or, going to be stomach, you can look into those particular tissues, which is not possible with micronuclei. And going back to micronuclei, you need to have a actively dividing cells to look at micronuclei, where in case of Comet assay, that is not required. So cell proliferation is required, and you can look at the many tissues in Comet assay. And you can also use this assay to explore mechanism of carcinogenicity in long-term rodent studies. So again, how we perform the acute combination studies. So this is similar to what we do in the micronucleus assay, where the top dose is 2,000 milligrams per kilogram. And in main assay, we have animals with 3 doses, vehicle and positive control. Plasma is collected, serum is collected to check liver activity. The organs which are collected for 3 samples for histopath, Comet slides, and tissue exposure. And we also collect bone marrow for micronucleus assay or peripheral blood. This is the parameters of DNA damage. We look at the percentage tail DNA, the tail movement, and tail migration. And if you need any more details on this, I can recommend you some papers which you can refer to. I think I'm running out of time.
Yeah.
And Comet assay is being used more and more to clarify the responses. Again, as per the ICH guideline, this assay can be used with micronuclei to demonstrate the clastogenicity. And there are few topics I want to just mention here. I won't go over the slides. For in vivo, this is for in vivo chromosome aberration assay or the micronucleus assay. For in vivo mutation assay, we use the Big Blue assay or the Big Blue assay and the PIGA assay. So both these assay look into the in vivo mutation. The main difference is for Big Blue assay, there is a guideline OECD 488, whereas for PIGA it's a new assay, there are no guidelines. Both have 28-day format. PIGA assay you can do only in blood, where in case of Big Blue you can do any tissues. And sampling is possible anytime in PIGA, where in Big Blue it's only in terminal, at termination you can do sampling. PIGA assay is much more less expensive compared to the Big Blue assay. And again, for Big Blue assay you need specialized animals, transgenic animals, where for PIGA you can use any animals. Thank you, Marcelo, for the opportunity.
No, thank you.
And if you have time for questions.
Any one or two at the most short questions?
I just have a quick one about,
Sure.
you have dermal exposure. You were saying about if you have no exposure, then your test fails. But have you seen a case where the rat skin is actually able to I mean, you would apply your chemical compound on the skin, but the rat skin then is successful at keeping that out, not letting it permeate?
We have one case. Actually, typically, when you do dermal exposure, you don't expect to go in your blood because it's a dermal exposure, so it should not go in your plasma. However, we have seen a case where we applied the compound dermally, and we got excellent exposure in plasma, which shows that it can reach bone marrow as well. But to your question, dermal exposure is used mostly for cosmetic industry, and in Europe or now most part of the world, in vivo studies are not used for cosmetic. You cannot use in vivo studies. Animal testing is banned. So we do offer something called as the 3D skin model, which is ex vivo. So we take the skin model and we apply chemicals on top of that and look at a micronucleus in that. So this is not a typical animal model, but it mimics all the in vivo aspects of skin to look at the gene tox endpoint.
But yeah, actually, you would expect the chemicals to go to plasma.
You would.
Only, I mean, if it's water, depending on whether it's water soluble or not water soluble.
Right, right. But you don't expect a high exposure in plasma with that.
Yeah.
Thank you.
Any other further question?
Sir, is this superior one than
Yeah. Can you use the microscope, please?
Yeah, this is superior one than DNA diffusion assay.
I didn't know.
There is a DNA diffusion assay is in a similar as the Comet assay. So I like to know that which one is superior, this Comet assay or this DNA diffusion assay?
Comet assay or
DNA diffusion assay.
DNA diffusion assay?
Yeah.
I don't know about that. Is that for gene toxicity? I don't
Yeah,
believe so.
genome toxicity.
I don't know about DNA diffusion
Okay.
assay.
I think it's not in use any longer.
No, there are no guidelines on that. So.
No. So there is a break. Yes, we will do it.
Okay, thank you.
I will move. Thank you very much. Up to you. 5 minutes. 5 minutes. Only 5 minutes? Okay, we will make a break of 5 minutes, and we'll return in 5 minutes. Because they asked to follow the schedule, and it was one break. If you want, we can continue. Up to you. Yeah? Okay. Oh, I cannot open it. Nope. Can you open it, please? Yeah. Thanks, Ed. How to put it in the whole system? I cannot see the icon. Where? Sorry. No, should be from one of these icons. Okay, let's talk about the use of animal and non-conventional animal models in gene tox studies. So far, the use of animals is extremely used worldwide. You can use such from small marsupials like the rat kangaroo up to the beluga whales. And the fact is that the use of animals in toxicological research dates back up to the 2nd century BC. And documentation of the use of the dogs and non-human primates in animal experimentation can be dated up to the 2nd century AD. For the first vivisection has been done. The more common animal models reported in the previous studies are the most often used in toxicological studies. However, non-traditional animal models be more appropriate or even better predictor of toxicity to humans due to a specific anatomical, physiological, pharmacological, or even metabolic characteristic that non-traditional animal models can mimic from humans. In addition, toxicity data from non-traditional environmentally relevant test species are used worldwide and required to support either environmental assessment, safety assessment of chemical products and waste, safety of environment, native or exotic species from environmental pollutants, including treated wastewater effluents, stormwater discharge, or chemicals, among others, but the most important group of these are the other chemicals that they are used in most part of the world and in huge amount. The vast majority of all toxicity testings is carried out in the context of regulatory requirements, as we have seen from our previous presentation, regarding the different parts of the world. Regulatory bodies or entities often emphasize the necessity of using one specific toxicity test to preserve either both the current level of human health and the environmental protection. Notification of all new chemicals placed on the EU at least market for the first time must be given to the competent authorities as of any members of the staff. Okay, these are some examples of the guidelines that have been talked in a previous talk. We were just talking about cosmetic and non-cosmetic ingredients. Testing requirements are not specified in the regulation, but it's very well known that it's in general requirement for safety which could be met by the use of animal or non-animal tests as well. A major influence of these developments has been the test guideline program of the Organization of Economic Cooperation and Development, which has developed standardized methods of testing methods of testing that are accepted in principle for all 3 members of the OECD members country. It has largely removed the need for testing according to different protocols to satisfy regulatory authorities in different countries, and accordingly has substantially reduced the total number of animals used for certain standard tests. One point that it should be taken into consideration that this organization also provides the focus for the introduction of new methods that replace, reduce, and refine the animal use. Change and revision has been extremely slow, but thanks God, during the last year, are many current initiatives within both the scientific and the regulatory administration that challenge the present practice with the aim of providing the same or even better results for human safety while simultaneously using or reducing the number of animals employed for each test. Now, I will show you some examples, at least that have been valid for our country, how we can use some native species, of course, including this non-traditional animal system, using the same endpoints that we have been dealing the whole morning together with you. In this case, I will give you some data and some protocols that have been used in one tadpoles of Rhinella arenarum. It's a conventional frog that inhabits most part of South America and lives near or inside the crops. So it's directly exposed to the most chemicals that they are using the crops. We have been using the tadpoles of this species because they are easy to handle under laboratory condition. In this case, the tadpoles were exposed to fluorochloridone-based herbicides, in this case 2 different herbicides containing the same amount of the active ingredients. They are reported for Argentina. And of course, we have performed all the classical tests for genotoxicity and ecotoxicology. First, the determination of the lethal concentration 50, the frequency of micronuclei, as well the induction of DNA strike breaks evaluated by the COMET assay. This is a classical protocol, as we have been discussing before. The treatment lasted for 96 hours. It was a continuing treatment after the end of the treatment, and as well as 48 hours, the frequency of micronuclei and the DNA damage evaluated by the COMET assay has been estimated, as I told you, 48 and 96 hours of the treatment. And the results that we can see that we were obtaining, it was very, very convincing that only one up to from the 2 of the formulation used by equal concentration, only one produced genotoxic effect when the micronuclei assay was performed, and indicating that most probably this is not permitted or related to the use of the fluorochloridone present within the technical formulation but the excipients that form part of this formulation. When the COMET assay was performed, we realized, and it has been also proved before and claimed before, that the COMET assay is a very sensitive assay for reporting genotoxic effects. And in this case, both commercial formulations were able to induce an increase in the frequency of DNA breaks. Okay, this is the aberration. This is another frog that also has been studied, but in this case, tadpoles were exposed to another commercial formulation of imidacloprid, and the endpoints were exactly the same as the ones that we have studied in our previous example. Here again, we found that the frequency of micronuclei has been only increased by only one dose after 96 hours of treatment, but not at 48 hours, while the COMET assay revealed that either at 48 hours as well as 96 hours, there was an increase in chromosomal damage measured by the extent of the DNA migration into the tail of the nucleoids. This is another non-conventional species that we have been using in our country since the last 5 years by one PhD student. It's a very small fish that inhabits all the crop area in our country. It is very, very small. It's a viviparous fish. It's very easy to handle and to acclimate within the lab. And this fish was used to analyze the genotoxic effect induced by some glyphosate-based herbicide formulations. And the endpoints were the determination of the lethal concentration, 50 and 86 hours, and the frequency of micronuclei. This was a typical protocol of a study. And the thing is that either In one of the formulations we use credit containing 48% of glyphosate. The frequency of micronuclei was increased either 48 as well for 96 hours after treatment. The Panzer gave similar results with the same frequency of the active ingredient within the technical formulation. I will go further and I will try to stress a little bit more like this. A complete knowledge of the toxic effect of the active ingredient is not enough in biomonitoring studies. A pesticide's toxic effect should be evaluated at least in ecotoxicology based on the commercial formulation available in the market rather than the active ingredient, and the deleterious effect of the excipients present within the commercial formulation should be not either discarded or underestimated. And finally, that a single biosec is not enough to characterize the toxicity of a pesticide under study or under evaluation. And finally, our short results highlight the importance and relevance of minimizing the impact on non-target living species exposed mostly to agrochemicals and other environmental pollutants. Thank you.
Any questions?
I want to be on time. Yeah.
Thank you, Marcelo.
I was running.
Yeah, that was very, very quick. We appreciate that, getting us back on time. I just had a quick question on the erythropoietic rate of blood formation in the actual tadpole, because the micronucleus endpoint and when you kinetically sample will be dependent upon that.
The problem is there is no any guideline for the tadpole how to be studied, when to be studied, and the length of the treatment. So far, the authors have not reached to any conclusion. Most of us, we are analyzing at least more than 1,500 mature erythrocytes per individual. And now becomes another problem, the number of individuals per concentration. Some old authors are claiming that 5 individuals are enough. In our hands, it should be analyzed at least more than 10, because the variation among individuals is great. And sometimes the variation can diminish or cover the real induction of any genotoxic effect. So there is no any pattern so far. The only EPA has decided to claim the number of individuals that should be analyzed, but there is no any specific guideline for this purpose. And the same happened with the treatment period.
Yeah, because kinetics would be interesting to follow it out.
Yeah.
If it's slow.
For some environmental pollutants, we have observed that the induction correlates negatively with exposure time, while for others, the induction is just the opposite. Some authors are claiming that a period of 96 hours is not enough to reveal, but in our hand, 96 hours is almost the maximum. If we extend the treatment up to 14 days, instead of having a dose-responsive relationship, we obtain nothing.
Thank you.
Okay. Is there any other question?
What was the route of administration of the herbicide in frogs?
It was dissolved in the water. This is another point. There is no any rule, so there is no problem to inject intraperitoneally. But since frogs at this stage environmentally are exposed when they are
Well, how do you,
living in,
how do you think that they're exposed? From water or from through their skin?
through the skin
Yeah, yes.
by the water who has been exposed. That's the only possibility, especially tadpoles of the second species. This frog is an arboreal frog. It's a very small frog, but with tadpoles quite big. And they have two different tadpoles. There are the normal ones lives for a couple of 20 days, 30 days, varying upon the temperature of the condition, but there is another possibility that the tadpoles has spent almost the whole year as a tadpole. But in this case, the tadpoles are huge, sometimes the double of the size of the one adult, but they are not useful for any study. No, they don't adapt into laboratory conditions. Yeah.
All right,
Okay.
to stay on time, I'm sorry, but for the sake of staying on time, we're going to go ahead. We're going to actually at the end allow for more questions.
Okay.
Next, we have Dr. Kamala Pant from BioReliance USA. He will be giving a talk on genetic toxicity screening assay.
Hello, how are you? He's here. Yeah, I know, but you know, I had some trouble for opening mine, so I would have the same trouble for opening yours.
Yes,
But how to put it?
easy. Yeah. Thank you.
Okay.
Hello, everyone. So my name is Kamala Pant, and I work at BioReliance. And Rohan and Leon have already given you overview of all the regulatory required genetic toxicology assays. So I am going to tell you about these screening assays. So why do we, well, this is the list of all the things that I'll talk about. I'll tell you what the rationale is for performing these screening assays. What are the different types of assays we do for screening? Little bit about the design, method, little bit, and evaluation criteria, and their advantages and limitations, if there are any. And then I'll discuss about the bacterial mutation screening assays, in vitro chromab, and in vitro micronucleus assays. These are the screening assays. Most of the time they are done. So what is the rationale? Why do we do these screening assays when we already have the GLP required assays that we have to do before we can submit for IND with FDA or the regulatory agencies? So the chemicals with positive mutagenicity results are frequently dropped from development. You have a Ames assay positive, and then it becomes really hard to overcome that positive response, and you end up either dropping it or doing so much extensive work. So positive results require disclosure and consent in clinical trials. And of course that leads to unfavorable labeling, because you have to tell that your compound was positive in mouse lymphoma assay or any other assay, and it may result in diminished market potential. So that's why pre-screening in at the time of drug discovery or lead optimization, it can weed out all these compounds which have slight mutagenicity in one of the assays. So this way, all the time and resources can be focused on promising candidates. And of course, it can facilitate efficient planning for the follow-up testing also. And screening or these miniaturized assays These can be performed with GLP. GLP is good laboratory practices, and they can be submitted for regulatory applications. So the non-GLP screening assays, these are used at early stages to select candidates for further drug development. Well, the advantages are, one is of course, it's lower cost. Second, it's very quick turnaround time. And the test article amount, not much is needed, very minimal amount. And these can be highly predictive of the GLP assays. And the designs can be customized based on available sample. However, there's a word of caution here that the design of these non-GLP screening assays should mimic the ultimate assay that we are going to perform for the regulatory submission as closely as possible for the best predictive value. So the types of screening assays. These can be miniaturized standard assays, for instance, predicted for the GLP regulatory results. The bacterial mutation assays. So these are Ames assay and Ames2 assay. So in the standard assay, we use 5 strains, whereas when we are doing screening, we may use just 2 strains, because these 2 strains give us good idea of if test article is going to be positive. Approximately 93% of the times, we will have the correct results. Then there are in vitro chromosome aberration assays, and we have the in vitro micronucleus assay. So first topic is bacterial reverse mutation, or the Ames assay screens. Now, I have the pictures of the plates, and I hope most people are familiar with the Ames assay. Ames assay is done in these 100 millimeter plates. They are this big. So we can miniaturize the assay and do that assay into 6-well assay, which is approximately 1/5 of the big plate. So that means the amount of test article also reduces by 1/5. Then we can do the same assay in a 24-well plate, where it's almost 1/20th of the big plate. So the amount of test article gets reduced. And then we have liquid microplate methods. Those are, we can use standard strains there, as well as the Ames2 strains. Ames2 strains were made just for the screening assays in Bruce Ames's lab. So the abbreviated standard assay, as I said, usually we use only TA98 and TA100. It can detect about 93% of the positive compounds. We can substitute or add strains if it is known that this particular class of compound gives positive response in, say, TA1537, we can add that. Assay is done with and without metabolic activation. And typically, we would do 8 doses. They start from 1.5 microgram per plate to 5,000. So then they get reduced in similar manner in the smaller assays. And we require about 90 milligram in the big plate, 18 milligram in the 6-well plate, and 5 milligram only in the 24-well plates for the screening assays. And we can do these assays with plate incorporation or pre-incubation method. And I won't go into method because Leon already covered this. So before we started doing the 24-well assay, we did a validation work, and we presented this data, and the work was done at BioReliance, and we tested a number of compounds in the big plates as well as in the 24-well plates. And from this slide, you can see wherever we have green, that's where we have the concordant results in the both. Where we have red, usually it doesn't match. But the reason is, this is compound to compound, it's 100% concordance. However, the concentrations may be a little bit up and down. So essentially, there was 100% compound to compound concordance, and 93% of the times the concordance was between concentration to concentration. And we use same tester strains as in the regulatory version, and of course can be justified to address the ICH M7. That is for testing of impurities in the drug candidates. So usually the impurities, you don't have so much compound. So you can use these miniaturized assays, and these can be used for submission for regulatory agencies. And these micro AIMS screens use about 94% less test article than the big plate assays. Now I'll give you a little bit overview of the AIMS 2 assay. Oh, and I had said that I'll give you, tell you pros and cons. So in this case, of course, all the advantages are there, less test article, quick turnaround time and all this. The disadvantage is you are testing lesser number of bacteria. You test about 1 billion bacteria in the big plate, whereas here it would be a lot lower. So that's the only disadvantage. Now, AIMS 2 screen. So these strains were engineered for base pair substitutions. They are TA 7001 to TA 7006 strains. So what we do is we grow all these strains individually. We measure the optical density, and we make them in and make this TA mix. They are mixed in the equimolar concentration, and the resulting strain is called TA mix. And then we use TA 98, which detects frame shift mutations. So these kits for doing the AIMS 2 assay can be purchased from Moltox, and we can use automated plating systems. And it needs about 6 milligram of test article. I just have a slide to show you the genotype of the AIMS 2 tester strains, all the base pair mutations there. So this is the assay. We grow the cultures overnight, measure the optical density, and then do the assay. In a 24-well plate, we would mix the bacterial culture, the test compound, S9 mix or buffer, depending on with or without S9, and the exposure medium. Incubate for 90 minutes, and after that, add indicator medium. Now this indicator medium has bromocresol purple. That's a pH indicator. So once indicator medium is added there, we plate from 124 well to 48 wells in this 384 well plate. And this is where the automated dispensing system comes in the picture. And once the plate is dispensed, we incubate the plate for 48 hours, and then we look at the plate. So since it has the pH indicator, what it does is, wherever there is cell growth, wherever the bacteria is growing, the pH changes, and you have yellow wells, and you just count them. And that's the whole assay. So it's very quick, easy, and easy to score also. So in this assay, I should give you the advantages. Again, advantages, it's only 6 milligram of test article. You are going to know any base pair mutation if the test article is going to cause. And for frame shift, we have TA98. The disadvantage is that there is no measurement of toxicity in this assay. Whereas in the regular AIMS assay, we look at the background lawn, and we know that, yes, the lawn is being reduced by this test article. Here, it's hard to tell, because if the color hasn't changed, that it could be because it's toxic, or it could be because it's not mutagenic. So that's the disadvantage. Now next is the in vitro chromosome aberration screen. And the design is very similar to GLP assay, except we don't do a dose range finder assay here. What we do is we would test a huge number, like anywhere from 9 to 15 pre-selected concentrations. And in our case, we use CHO cell line or blood lymphocytes to do the chromosome aberration assay. And we collect the metaphages by mitotic shake-off, meaning we will shake the flask and only the dividing cells are there. So that way when the slide is being scored, all these dividing metaphages are there, all the metaphages are there. And the highest concentration is scored based on 50 to 60% cytotoxicity. And that's with the relative increase in cell count in the CHO cells or by mitotic index in the human peripheral cells. We scored 3 dose levels. Positive and vehicle controls are always included. And we don't do chromosome counting in the metaphase. What we do is we look at it, and if there are approximately 20 chromosomes, we would score them. Anywhere from 100 to 200 metaphages are analyzed per dose. Now, aberrations are not classified. They are simply categorized. So we will categorize them as normal, aberrant, polyploidy, or endoreduplication. So I have the picture here. So the first screen here, it would be just for where I'm showing the breaks and exchange, that's aberrant cell. Then I have polyploidy where the number of chromosomes has doubled or tripled. And then I have endoreduplication here. So then here are the advantages and disadvantages. Of course, significantly less test article is required. That's the advantage. Disadvantage is we are scoring fewer cells and reduced statistical power because of that to detect weak response. And we may not evaluate all 3 treatment conditions that are regulatory required. That is, in the regulatory required GLP assay, we have with and without S9 for 4 hours, and then we have 20 hours without S9. So those are the disadvantages. And now I'll discuss the in vitro micronucleus screen. And this is also very similar to the ChromoAb assay. So in our case, we use either CHO cell line or TK6 cell line, and we can do the assay with the human peripheral blood lymphocytes also. So again, we do 4-hour treatment with S9, 24-hour treatment without S9, and we harvest the cells at 24 hours after the start of treatment. So approximately 1.5 to 2 normal cell cycles. And cytotoxicity is based on the cytokinases blocked proliferation index. And Leon went through this. So we add cytokinesin B and get these binucleated cells, and we can score them. Normally, we score 500 to 1,000 binucleated cells and analyze, these are per dose, and see how many micronuclei we have, compare them with the vehicle control. And the doses for scoring are selected based on 50 to 60% cytotoxicity. So here I just have pictures of some micronuclei. The first one is GIMSA stained, and there's 1 micronucleus there. And the other one is acridine orange stained, and lots of micronuclei there. In our case, we did the validation, and we reduced the volume of treatment from 5 ml. See, the GLP required assays we do in 5 ml of blood and media suspension, whereas in the screening assays, we can do it in 1 ml. So similar to Ames validation, we found that we had 100% concordance between compound to compound. Positives were positives, negatives were negative. And the miniaturized screen had similar sensitivity to 5 ml HPBL micronucleus assay, about 95% correlation between concentration to concentration. And 1 ml micronucleus screen used about 80% less test article. So we needed only 60 milligram versus we need 300 milligram when we are doing the 5 ml assay. And this work, we did this work at BioReliance too. So here's the slide. it shows that wherever we have same results in both test articles, and the red is for wherever we have different results. So the different results were only in the concentration, and that was because sometimes it's more sensitive in the 1 ml versus in the 5 ml assays. So the disadvantages and disadvantages. Again, it's same. It's a lot less test article is needed, and we score less number of cells. So that's the disadvantage, and reduced statistical power. So may not detect the weak response. And of course, we may not use all 3 treatment conditions. That's another thing. So then there are a whole lot of other screening assays also. So virtually any regulatory assay can be scaled down to conserve test article time and money. So fewer dose levels, replicates, strains, animals, we can reduce all of them, and fewer cells scored. So we can do CHO HPRT assay in the screening mode, mouse lymphoma assay, in vitro COMET assay, in vivo micronucleus assay. What we do in vivo micronucleus is, instead of doing 5 animals per dose group, we will reduce the number of animals. And cell transformation assays also. In cell transformation assay, there's a newer version of assay called BHAS42 assay, and we can do that assay in the 96 well plates. So that's great for screening. So in conclusion, these miniaturized screens correlate well with the corresponding regulatory required assays. The closer the design, the better the correlation, of course. And miniaturized screens require substantially less test articles, so they can be done at the time of discovery or lead optimization. And earlier screening to identify genotoxic chemicals earlier in development saves valuable time and resource, because then the compounds which are not going to give you positive response can be developed. Thanks, and if you have any questions.
Any
Yes.
question?
Well, I'll try to shout. How linear is the response in cell growth and pH changes that you use in the Ames assay screenings?
How linear, you mean,
If you double the number of cells, can you detect that by pH change? Saturated growth.
right, so I don't know about that one. However, you see bacteria grow every hour,
Yeah.
and we are waiting 48 hours.
Okay.
So in 48 hours, they have gone through so many cell cycles, and we are seeing them after that time. So, yeah. And usually it's very nice. I mean, scoring is great. You just look at the plate, and there are these yellow wells, there are these purple wells.
But nothing in between?
Well, there may be in between too, but usually they have to decide one way or the other. Yeah. Okay, thank you.
Okay, thank you. Okay, let's move to the last participant, Dr. Daniel Roberts from Bristol Myers Squibb Company. Welcome, Daniel.
Thank you, Marcelo.
I hope it is well.
Okay, hi, I'm Dan Roberts from Bristol Myers Squibb, working there for about 5 years. I'm also a PhD candidate at Rutgers. Because I didn't have enough to do, I figured I'd go back to school. I'm going to be talking to you today about how we use all these assays to actually support pharmaceutical development. So a lot of you are wondering, these gene tox assays, you've heard about a dozen of them. Well, what's the point? Well, in my industry, the point is to predict the rodent carc assay, to predict the outcome in order to get into humans faster. So the first thing you have to consider is all the modalities that actually exist as potential therapeutic agents. And I'll be focusing on small molecules because it does fit well with what's been described. There's also antibody therapies, and if you ever have a chance to dive into that, it comes down to what the antibody, how the antibody is generated and whether or not unnatural linkers are used to attach a cytotoxic payload. Because then you have to do a whole risk assessment on the linker itself. Proteins and peptides, they get kind of hairy because if you're using the salmonella strains that are oxytrophs, then you're adding in histidine or tryptophan as your test article. You could see concentration-related increases that are just due to the extra histidine in the mix. So I'm not going to touch that. Then there's this new emerging field of therapies with oligonucleotides. And so far, the 2 dozen that have been tested are completely negative in the gene tox battery. So there's a lot of debate right now on whether or not we even need to test those. So considering that small molecules is the most worked out, and you've heard about all the assays that are expected from the health authorities, I'm going to focus on that type of modality. There's also multiple therapeutic areas. So for oncogenics, you really only need gene tox for marketing. You know, if the patient already has cancer, secondary cancer is very likely. So the health authorities want to see this new potential medication quickly go to humans to see if it's efficacious prior to wasting time doing extra gene tox work. There's also orphan therapeutic areas which have different type of filing requirements and patent lifetime. And then common things like virology, going after HIV, HCV. And Bristol Myers has a long history of that. But this will be a focus on the non-oncogenic common medical conditions. So the main goal will be to describe how we use all the tools you've heard about to assess risk after hazard ID and to enable and progress these small molecules into and through the clinic. So what is our perspective? We kind of hit everything in pharma. You have to be worried about accidental environmental release. However, that's mostly nowadays with reagents because the actual pharmaceuticals are manufactured at site. And so the REACH regulations that touch the big reagents that are shipped to each site impacts the environmental kind of safety. There's also worker safety, which we just absorbed 3 occupational toxicologists in our group. And they do a lot of what they call banding of chemicals internally. So you have to keep your workers safe. If you're mixing up a vat of cytotoxic payload, you don't want to breathe that in all day. And depending on how hazardous these reagents are, it impacts where you can manufacture drugs. And that can cost a company a lot of time and money. But of course, our main focus is patient safety. We're trying to build medicines. We want to make sure that they're safe. You don't want to take medicine for diabetes and then wake up with retinoblastoma. Not really good. So when you give an industry talk, I've been told I have to go through this. Basically the pipeline. And instead of giving you the general pipeline slide that you've probably seen on Google 30,000 times, I thought I'd break it up into really developmental goals from a pharmaceutical perspective and then the gene tox work that accompanies these goals. So starting with discovery, the real overall goal from a medicinal chemist point of view is to identify efficacious candidates. And then, of course, do basic hazard ID. For the gene tox work, we do exploratory studies in-house. For mutagenicity, we actually screen in the larger version of the Ames assay, even though we heard about nice miniaturized alternatives. Reason being is because of the cell number. We want to test the full amount of cells. If compound is limiting, you could do an SOS chromotest, which is kind of a repair assay and isn't 100% predictive with the Ames assay, but does an okay job for triaging. The other exploratory study we would do is a classigenicity endpoint, which in our hands is the micronucleus assay because we can automate the scoring by flow cytometry. And then in silico analysis is now quite a useful tool, especially for the chemists early on, even though most chemists nowadays know what is genotoxic and what isn't. Although lately we've had, of course, more examples. Preclinical, and I think you've seen this Ames plate picture like the 3rd time now. I think we're all sharing the same picture. So once an early candidate nominees are basically brought to candidacy, that's the internal lingo they use, they pick 5 compounds for a therapeutic area. So let's say cardiovascular. Now, of course, you go into your tox work, you set your safety margins. And we put together the IND package with the toxicologist to help go to first in human. At this stage, we're also trying to look at any genotoxic impurities. That is going to be the future of gene tox, I believe, is the impurity work, because for every one compound, you may have 20 reagents. For each reagent, you may have a certain purity. So there we do the same studies we do exploratory, but we simply do them with GLP so we can submit them to health authorities. Once you get into the clinic, the overall goal, of course, is to establish human efficacy and safety and enable the new drug application. Gene tox work there becomes quite interesting. There you're looking at unique and disproportionate metabolites. You're looking at the reagents in order to make sure that they're safe for your workers, and as well as to select manufacturing sites with appropriate engineering controls. And then once you actually get to the market, to improve efficiency, a lot of times they'll change the reagents or their sites, which then they just bought a reagent that has a genotoxic impurity, and all hell breaks loose, and then we step in and try to save the day. So it keeps it fun. So drug discovery screening test for actual pharma. The combinatorial chemistry flop basically forces us to still do rational drug discovery. And it works relatively well with the in silico tools. When you have an in silico tool telling you that there's no structural alerts for mutagenicity, the big one in the middle, for mutagenicity, if it's negative, we go through the Ames assay and then do a classigenicity assay. However, if it is positive, we would still follow it up with an Ames assay if it's a drug that is important to us to develop. If either of these are positive, we generally stop development or triage. So gene tox folks can either be your best friend or your worst nightmare if you're a chemist. We would either stop development or triage, meaning up here we would do structure-activity relationship assessments in order to try to get around this genotoxic hazard. Best case scenario is screening's negative, and then you proceed to the IND GLP studies, which you've heard about. And at this stage, especially if there are hazards, a lot of toxicologists basically are like this cartoon. Like, well, what are we going to do for the IND? Because they're used to their tox work, not gene tox. So there's three main buckets here. First is no hazards. So there, as you've heard briefly, there is an option where you do 2 in vitro assays, the Ames and a classigenicity assay generally. And then you can piggyback a micronucleus bleed onto your 1-month tox study. So that reduces animals, and since there was no hazard identified in vitro, there's really no risk. You're just basically setting safety margins. If you do have a classogenic hazard, however, you have to still do the bacteria assay to look for mutation, but then as Rohan mentioned, you kind of do hazard ID in vivo. So you end up dosing rats up to much higher doses than are needed, which drives a lot of therapeutic area heads nuts. But the idea is to look at classigenicity in at least 2 endpoints to ensure that there's no hazard in vivo prior to going to humans. If there's a mutagenic hazard, you're done. Usually you park the molecule or build costly weight of evidence, which I'll go through. Bristol's not in the habit of doing that, but other larger companies with bigger budgets probably are. So classogenic follow-up, you have to demonstrate lack of risk, so health authorities want to see this. You could do reproducibility in another cell line, maybe human versus rat, as has been mentioned before. However, you'll likely still have to go in vivo. So we would do a rat study with at least 2 tissues evaluated, the micronucleus, which you've heard a lot about today, and then the comet assay, alkaline version, and duodenum and liver, because that is where we consider the first site of contact due to absorption when you give a drug orally, and then metabolic capacity in the liver. Worst case scenario, if we had one compound where I think micronucleus was flat and comet because of how sensitive it is, a very weak marginal increase that didn't quite meet our positive response criteria, but to be cautious, you still go ahead and run the 6-month CARC study. So it's a good point too. I'm sure most of you are familiar with ICHS1, but the whole 2-year rodent CARC study is kind of going away. This transgenic 6-month model, the HRAS mice, it's just as sensitive but more specific, and it's being used now to escalate timelines into the clinic. However, we try to avoid doing that until we absolutely have to later on in the phase 3 clinical. So mutagenic follow-up, again, we wouldn't necessarily do this, but if you have an Ames positive result, it could be due to a bacteria-specific metabolite. And there is an app that needs updating. So the Ames bacteria have nitroreductase enzymes that can actually reduce nitro compounds to genotoxic intermediates. And we had an example of this with a bunch of reagents. And you can buy nitroreductase deficient strains and run them side by side, and you can see an amelioration of the mutagenicity, which indicates it's a bacteria-specific finding. And that, of course, opens up strong debate with the, gut flora or gut fauna. But in the interest of time, what you really would do with an Ames positive result is go into a different type of transgenic model using the LAC-Z transgene type models. You heard briefly about Big Blue, and what it ends up with is if your LAC-Z is mutated, you have a faulty beta-galactosidase transcript, and you can't metabolize X-gal. And then you count these blue and white colonies to get a mutation frequency. The nice thing about these transgenics is you can do any tissue. So if your compound is working in B cells, you can look at the spleen as well as other tissues. PIG-A gene mutation assay, which was briefly mentioned, currently it's approved for genotoxic impurities, but not active pharmaceutical ingredient. I have a feeling this will change. The OECD was just kicked off in April of this year, so we have a few years to finish getting that ready to go. But that is a simple blood test, and we heard earlier this morning how great simple blood tests are for adding on and using them as biomarkers. And again, if you want to just throw all your cards in and just say, I'm done, do the 6-month CARC, you could. It's usually costly, and with how fast decisions have to be made, you generally don't have time as a luxury. So antigenic follow-up, and I only have 2 more slides. I'll try and move as quick as I can. This was mentioned with the in vitro micronuclease test, and we use this all the time in our screening platform so that we can detect antigenicity. And if you're screening with in vitro micronuclease, for human cells you can use a double-strand break marker or a mitotic/hyperdiploid marker in order to understand polyploidy and antigenic induction. In rodent cells, which is the platform we currently utilize, we can simply just quantify hypodiploid nuclei in our lyse nuclear spreads. And it's pretty well established that with rodent cells, it's very indicative of antigenic mechanism of action. So the gold standard, of course, is to label the kinetochore proteins or actually do some FISH to hybridize to the centromeric regions of DNA. Here's a cell with a small micronuclei. This is biconfocal, so it's kind of out of plane, but there is a kinetochore positive micronuclei with the merge. And the reason this is important for pharma and really for any type of risk assessment purpose is this is indirect genotoxicity. So antigenicity is not DNA damage as a primary cause, or effect rather. It is a secondary effect. And so we can go and do a bone marrow micronuclease test in rat and be able to set a good safety margin to go to first in human. So clinical progression, this is a part of the work that does impact genetic toxicologists if they're working in pharma. I currently don't do much of it, but I thought it was worth mentioning so that you have a well-rounded idea of what we do. Human metabolite evaluation, once you're in the clinic, is important. If things do arise and they're unique or disproportionate, unique meaning human only, disproportionate meaning over 10% of parent, then you have to do some follow-up work. If it's unique, it's extremely hard to qualify that in a rat study because the rat may not make that metabolite. So you may have to spike it into the active pharmaceutical ingredient prior to dosing. During this time, they've also done long-term stability studies, and you'd be surprised how many times the degradant comes up as being a genotoxicant. So those are usually qualified as well, and it's usually in the terms of a 1-month tox study, and we just add on endpoints. So controlling exposure to GTI, as I mentioned, is very important. Impurities seem to be popping up left and right nowadays. And then the REACH regulations and occupational environmental safety. This is where there's growing interest in GeneTox to help kind of build this as more of a formal intelligent risk assessment, because kind of with most environmental and or general population hazard ID, it's kind of a checkbox exercise. With pharmaceuticals, there's been a lot more work, probably because it's part of the economy, right? It's big business, where they've made sure to, with good science and regulatory approval, kind of change how you use the GeneTox tools and pieces in order to bring something that might be life-saving to market. So in summary, we support drug development and as needed, which is needed for enabling first-in-human studies. GeneTox use for patient safety, but also worker and environmental safety. In-house screening strategies that we do at Bristol does help us rapidly identify any hazards, and we can quickly triage them in-house. And this is nice for any pharmaceutical company because it hastens therapeutic area development. However, there's a lot of times now people are outsourcing a lot of their screening strategies and most of their GLP work. Bristol seems to be hanging on to their GeneTox folks, which I'm quite happy about for now. So the big take-home message, which shouldn't be a surprise to toxicologists, is positive in vitro test results really only identify a hazard. You have to go in vivo to actually look at risk. And usually the risk for pharmaceuticals, that's where all the real work and importance lies. Because if there's a hazard but it doesn't translate into a real risk, you don't want to prevent an actual life-saving medication from coming into the market. I'd like to thank some folks, my boss Laura and Jim, they reviewed the slide deck. Marcelo, thank you for setting this up. And I'd also like to thank my family, because right now I'm supposed to be on vacation, but I got the green light to come here and spend time with you. So, thank you.
Any question?
Any idea on the cost of this 6-month card replacement as you talk about?
It's a good question. I mean, it'd probably be at least 1.5 million. I don't have those numbers in front of me. And with how much they fight and, you know, they go to like 10 different contract research places, I'm sure they can get a good deal. But yeah, we generally don't do that until we have to in phase 3. Like we wouldn't use it for triage, but there's stories out there in the literature where other companies have, and they're actually quite interesting. So.
Any final question?
All right, thanks.
Thank you.
Okay, so just quickly, there are speaker certificates that we'd like to give out to all of the speakers. So if we could have all 5 of the speakers from this session come up. My program actually says you're supposed to help. Can you help me give these out? Just no, these. Come up here for a minute. Just come help me. All right, so if I can get all 5 of you up here. Omics would like to give you a certificate to thank you for your participation, so if you can actually help me hand these out, I would appreciate this. So Leon, if you want to give it to Leon. It's like elementary school, right? Raise your hand, you're here. So thank you very much. You're welcome. And put you right in front of the light, right? Marcelo, you're next.
Okay, thank you.
Sure, Karen too. Kamala. Thank you. And Dave. Thank you very much. Okay, so that's it for this session. But then we have another one that's supposed to start right now. So.
Right now?
I think it's a break.
No, it shouldn't.
No, there's
No.
not a break.
Yes, there
There's
is.
No, there's not. There's a 12 o'clock session.
Oh.
And then it's a 1 o'clock lunch. That's what the program says.
It does.
Yeah. So, all right, we're gonna make things up on the fly here a little bit. I think we can do this. They want us to do a group photo, which I think we can do on the way to lunch. Is everybody okay with that? We'll just sneak by for a group photo on the way to lunch. And then there are 3 talks, I believe it's 3, that are scheduled before lunch. And then lunch is supposed to be 40 minutes, which seems a little short to me. So I propose, and this is me making a suggestion, I propose we do 2 talks before lunch and then shift the other one to after lunch so that we don't miss our lunch slot in the other room. Is the 3rd speaker opposed to that? I don't know who the 3rd speaker is. It's you? You're okay doing it right after lunch? Okay, and then we'll shift a little bit there. Is everybody okay with that? So let me go ahead and invite the chair and the co-chair of this next section up here to be able to do theirs. I think you guys are done, but you're welcome to stay. I don't. So if we can have Dr. Kumar come back and Michael Morgan, if you'd like to join us to chair this session. That's all I've got. You're off.
the hand. You're welcome. Where did it go?
Is this it?
Yeah, that's it.
Wait, it's not quite turned on yet.
Okay.
All right, I guess our first speaker.
Let me just make sure I get it.
Dr.
Go ahead.
Stryford. Okay, what happened?
Got it.
Just a second, wait a minute. Okay. All right, I think I'm ready now.
You good?
Yeah.
We're working on it.
Is it in the right place? Yeah. No?
Let's try this. You want presenter mode, right?
Yeah. I want to get it this way.
You go however you get it.
Oops. Let's see. Let's try it like this. What about if I go here?
You should be fine.
See if it goes. Wait a minute, we're on number 1.
You're good.
You
You're
did?
good.
Okay. Nope, it's not working. Wait, no, it's not advancing. Mm-mm.
Yeah, there you go.
Where were you? Where did
Just
you
push
go?
the space bar. And it'll
Not
advance.
this?
Anything is fine, but the space bar
Oh,
will work
okay.
for you.
Okay. My name is Kim Stryford. It's nice to see you all. This morning I'd like to visit with you a little bit about a hypothesis that I've come up with about how oral contraceptive use is linked to the development of autism in children of the women who take oral contraceptives. And so in order to understand how I came up with this hypothesis, I think we need to look at autism itself, oral contraceptives, and then I would like to describe to you how oral contraceptives are endocrine disruptors, and they're capable of initiating harmful effects, both in the endocrine system and also in the developing neural system of children. Do any of you here have an autistic child, or are you related to one? No? You might wonder then how I came up with this. I was working with children who are on the spectrum, who have autism, and I was working with them with equine therapy, and I noticed how unique they all were. All of them were different. Each child was different on the spectrum, and they had a lot of different attributes to them, and so I became very much interested in them. So we do know that it's a complex neurodevelopmental disorder, and also that there are social impairments and communication difficulties, and sometimes there's repetitive behavior and stereotypical types of behaviors. For instance, the children at the barn were unable to communicate. They wouldn't make eye contact. They wouldn't look at you. There was repetitive behavior with their arms flapping and the toe tapping. Sometimes if they did speak at all, they would just repeat something that they'd heard. So we know that they have social impairments. The other thing we know is that there's also 1 in 68 children now, and that the prevalence is increasing. Since 2002, the CDC reports that the prevalence has increased by 120%. So scientists have offered many explanations for the increase. They've offered absolutely everything under the sun to explain. Now, the potential explanation for the increase falls into 3 categories. The first is identification and screening and diagnostic criteria and awareness. Some people say that it's an epidemic of just new diagnosis and awareness. Other scientists have offered that it's a genetic variation. So they say that perhaps it's an addition of a gene, a mutation of a gene, DNA methylation of a gene. That's been offered up. And the last category that people have offered is that it's an environmental problem. Here's just a sampling of environmental explanations that people have used for the increased prevalence. Pre-birth weight and low birth weight, multiple births and breast presentation or a cesarean delivery, assisted reproductive technology, parental age, socioeconomic inequalities, parental exposure to occupational chemicals or pesticides, living close to freeways, folic acid, the MMR vaccine, excessive multivitamins, acetaminophen, dietary factors. All these things have been offered, and yet we're not any closer to understanding some of the causes of autism. Researchers are now starting to look at changes to the brain, and there's evidence that underlying modifications in the brain occur long before the time when the symptoms are even apparent. It's now been almost 60 years since scientists identified and described autism, and still it's an unsolved mystery. We still don't know underlying causes for this disorder. But scientists believe that it's a complicated interaction between genes and the environment. I agree with those that think that the increase in prevalence is real and not just an artifact. And so I was thinking to myself, over the past 40 or 50 years, what have we women been doing? What have we been doing to change our babies' brains? And I thought about it and I thought, you know, I think I've got it. The increase in using oral contraceptives and other hormonal methods have increased over my lifetime about the same rate as the increase of autism. And so that's sort of where I started with my thinking about this and researching it. Strikingly, as the prevalence of autism has risen, so has the usage of oral contraceptives. In '62, about 1 million used oral contraceptives, and to date, 11 million women in the United States use oral contraceptives. 82% of all women who are sexually active and using contraceptives, 82% of women have used oral contraceptives. Now, they're effective because they have two parts to them, two compounds, an estrogen and a progesterone. One thing I found that was interesting was that a lot of people, women especially, don't understand how oral contraceptives work. The nurse midwives did a study and found out that women didn't understand efficacy or how most contraceptives, let alone oral contraceptives, work. And we also found in my study that there were also medical students and interns and doctors that also didn't understand. So I think that there's some misconception about this pill that we think is just like aspirin. So here's how they work. First, the progestin suppresses ovulation, the release of the egg. And then next, the estrogen augments the action of the progesterone and stabilizes the endometrium. And the primary mechanism of action of the oral contraceptive is to inhibit the development of the ovarian follicle and to prevent the release of eggs. All right. Think about this. Progesterone negative feedback reduces the pulse frequency of the gonadotropin release hormone released by the hypothalamus, which decreases the secretion of the follicle-stimulating hormone, which we call FSH, and it significantly decreases the secretion of the luteinizing hormone by the anterior pituitary. The diminished levels of the FSH inhibit follicular development, and they prevent an increase in the circulating blood levels of the estradiol. So the progesterone negative feedback and the absence of estrogen, positive feedback on the luteinizing hormone secretion, inhibits the LH serve in mid-month and inhibits the follicular development and prevents ovulation. How many women knew that? I mean, how many people know that it is that complicated and that sensitive of a process? I just think that women aren't aware of that. The other thing that I found out is that most modern formulations use the synthetic estrogen, which is called ethinyl estradiol, EE2. And that is a known endocrine disrupting compound. And it's capable of causing harmful effects to the endocrine system and also to the developing prodigy. Endocrine disrupting compounds have the same attributes as hormones. They're able to be active at very low concentrations. They're able to bind to receptors at very low concentrations. They have the ability to interact with multiple hormone receptors concurrently. They can work simultaneously to create additive or synergistic effects not observed with individual compounds. And they're often not feasible to extrapolate low-dose effects from high-dose effects. So the mimicry was what was interesting to me, that these compounds are actually able to mimic an androgynous hormone using the same mechanisms. The World Health Organization recently issued the State of Science of Endocrine Disrupting Chemicals in 2012, and they said that we're just beginning to comprehend the mechanisms through which endocrine disruptors exposure can modify the development of specific tissues that lead to increased vulnerability of diseases and disorders. What was most interesting to me was exposure timing. I was curious to what would happen, what's happening to the follicles and the eggs if oral contraceptives are designed to disrupt a woman's normal keep her from being pregnant. Then what's happening to the follicles or to the oocytes? Are they repeatedly assaulted every month? Does the assault modify the oocyte or the egg, or does the egg die? That became interesting to me. I also wondered if perhaps after fertilization. So we know that the pill is effective. Only 1 in 100 will get pregnant if they take it exactly as prescribed. But 9 out of 10 will get pregnant if they miss a dose or they don't take it as directed. And that means, if you're talking about 11 million women, that means it's possible for about 100,000 women to get pregnant. And then the embryo itself and the developing fetus might be exposed to EE2. And lastly, I'm interested in the transgenerational transmission of modified epigenetic programming. And this just means that perhaps it doesn't stop with one generation, but is transmitted to the next generation and also with concurrent use in the next generation. So, oral contraceptives are marketed as being safe and effective. And I think that the safety part of it, when the FDA was approving them, was basically based on efficacy. And now I'm asking about, they might be safe for the woman using them, but are they safe for the offspring? In 1966, there was an FDA report that was put out about oral contraceptives. They broadly looked over all of the safety and efficacy issues. But what was of particular interest to me was in the appendix. One of the members, Roy Hertz, gave a warning to all of us about oral contraceptives. And he presented the exact same question I'm asking. What happens to the oocyte? What happens to the ovum when it's exposed every month to oral contraceptive, and especially to this EE2? He said that we would have to study 100,000 children to be able to understand what the implications would be. And as of yet, we haven't done that. To date, there's a real lack of comprehensive research into the potential neurodevelopmental effects of oral contraceptives. I went through all the PubMed, I went through all the annual reviews, 30,000 annual reviews. There's nothing written about oral contraceptives and autism or autism-related disorders. I found one study, and the one study was done by a woman by the name of Lyle. And she had 66,000 nurses that she looked at, and their risk factors for different hormonal lifestyle changes. And of the 66,000 that she looked at, the one thing that stuck out as significant was use of oral contraceptives and the duration of that. So that was a little bit of epidemiological evidence for me. I'd already come up with the notion of prevalence, and then this epidemiological evidence has come up as well. So in support of the hypothesis, there's a temporal correlation between the use of oral contraceptives and increased prevalence. To date, there's no definitive cause or contributing factors for the increase. Oral contraceptives disrupt the endocrine system. Oral contraceptives directly and deleteriously affect both the ovaries and the ova. Likely effects of oral contraceptives on progeny are open question. And we haven't done the called for further study and controlled follow-up about the transgenerational effects that could be possible. This multi-hit model, I'm sure you're all aware of this. It was developed by a person who is studying cancer. His name is Knudsen. And it's a multi-hit because it describes how this might occur. I'm saying that the oocyte or the ovum might have the first hit and then later be assaulted by either a genetic factor, or an environmental factor, or an epigenetic factor. So risk factor is what I'm talking about, and it might be combined with other risk factors. The last slide is another recent hypothesis that I've just published in Medical Hypothesis, and this is presenting an epigenetic model. In this hypothesis, the steroidal synthetic estrogen, EE2, binds to and activates the estrogen receptor, which then binds to DNA, increasing DNA methylation at many sites across the genome, including the normal target genes of the estrogen receptor. But moreover, methylation of the specific estrogen receptor gene silences and decreases the gene product, the messenger RNA, which decreases estrogen receptor protein and its signaling abilities in offspring. So at this point, I've concluded that there's some epidemiological information and evidence, although we need more. And I've also come up with this model, which could be tested in animals. And so I'm hoping that I will get some help with this, and that people will start to look at animal studies and try to get to the root of whether or not this particular compound is initiating these adverse effects. Any questions? Yes?
When you think about it, it's absolutely amazing that there have been no work on this. We're in the wandering around for the last 60 years, and nothing has been tested properly.
Isn't it amazing? And most of the mechanisms of action are old. Most of those, the pill and all the things that women are given are old technologies. 1988 was the last year there was any funding for contraception, 1988. And just the fact that people don't know about it, but what I gathered and why I kept studying so much about it is we think it's as safe as the pill. It's a kind of buzzword, this conventional wisdom that it's safe, but there hasn't been study on it for the offspring. No study. So
a very interesting talk and a very interesting hypothesis. So I wonder, will you perhaps design a study to prove all these your suggestions?
As far as the epidemiology goes, I've suggested there's a large cohort in Norway, mother and birth cohort. It's a very large cohort. And the people in Columbia, Columbia University, are heading it up. And I've talked with them because they have the information that they could do an epidemiological study. As far as this goes, I just published this. As far as this goes, I'll have to have someone make a mouse. I'll have to have a mouse model, and you know, that's extremely expensive. But I think it can be done. Did that answer your question?
Yes, thank you.
So the difficulty with a mouse study, the numbers that you're going to need, and the animal with people, how are you going to do a really big study?
Yes.
I mean, humans are wandering around.
I know. So that's why I said the epidemiology, because this is five studies right here. This is five studies. But the epidemiology would be the best. And the nurses study, I already found that there was a correlation in it. And I've contacted them as well. That's out of Harvard, the nurses study. And so again, here I am, I'm trying to.
What about the FDA?
Yes. What about the FDA? So what about the FDA, right?
And perhaps women don't, or aren't all would be very concerned if they realize that the fact that you provided a great help to the way we lived, released women from all sorts
Yes.
of things.
Yes.
So,
It's very complicated. However, I wonder, it's one thing about a woman taking the risk for herself, but I can't imagine any woman saying, I'll take the risk for the child, right? So I think because the FDA has said they're safe and effective, and remember at the beginning it was about efficacy, it really wasn't about safety because there was blood clots and all sorts of things. So I think what happened is they didn't do the study that Hertz said to do. I mean, he is a Lasker winner and an NIH professor, and he told them that it would take further study and follow-up, but it was in the appendix. It was at the back of the FDA report. And when you look now, the FDA says on their page, they even say that oral contraceptives wouldn't pass muster now. They wouldn't. So, but now they've become lifestyle drugs, and they're just,
but like aspirin wouldn't pass the teratogenic effect.
Exactly.
It's now being used for cancer. It's being used for heart. It's now come back as the wonder drug.
I know. Yeah. Further study.
So what's the situation in Norway? You said, because
Okay.
that's what I'm interested in. In other countries, oral contraceptives are not necessarily as
That's right.
prevalent as here. So
Okay, so what I was talking about with this mother and child study, huge. In places like Europe, you can get a lot of a population because they have health care where you can look across. You could look across. There's this recent study. It's called MOBA, and they have enough women and autistic kids. So there's two parts of it. One, women and all the risk factors they took, age and whether they smoked and whether they didn't smoke, the women, but they also have the children. They have all the information with autistic children. So they're able to make correlations between the two. And they can look, and they have all kinds of pills and hormonal things that they've taken or what kind of birth control, and they can just easily match it up. They can just easily match it up and show out of this population whether or not there's an epidemiological risk.
that might have been already been reported, right?
No, I published that. I just published that.
The rate of autism in other countries, not just in the United States, in the
Right.
United States versus the rest of the world.
Okay, so the rest of Pistula and Pistula published across the world. So that those people, just same time I did, they published the increased prevalence rate all across the world. And in some places it's low, and in some places it's high. I just focused on the United States. I just focused on, because we are the greatest user of them, of oral contraceptives.
Yeah, you have been talking about the relationship between the binding of E2 with specifically into DNA. Do you have any clue what would happen with those 46XX embryos in which one of the X chromosome is already inactivated by methylation?
Right, and so what I focused on was that cancer, I use the
Yeah.
cancer models. So all the cancer models showed to me that this mechanism would work. And then I had to look to make sure that autoregulation, that was another piece. I mean, there were several pieces of this. That's why I said there's about five projects here. And the last part was that I found out that they're not silenced. The estrogen beta is not silenced when it goes through the global DNA methylation.
Mm-hmm.
That was what was most outstanding to me. Because normally, when it would be go through the global methylization before the embryo, but it doesn't happen. So it can be carried forward. Yeah.
What about the EU, the MOBA study that you're on about? Because I was involved about 3 years ago in a study which also MOBA came into.
Mm-hmm.
And this is where we were trying to find out that fathers can be just as,
Yep.
they all think that everything happens from the mother. It doesn't, because the father can pass on as much damage to the offspring as the mothers. And so they should wait for 3 months for the cycle to go through if they want children without damage passed on from the dads.
Mm-hmm.
Now, in that was EU funded, there is another group getting some stuff from the MOBA group.
Mm-hmm.
So have you written to the European Union?
I've only gone through MOBA, and through Poland is also doing a study. But Poland is also hooked up with the Norwegians at MOBA, and MOBA is controlling almost all of that over there now at this point. And I have been in touch with the Norwegians, but not with the EU.
Well, the EU has got but it has to do the whole of Europe. It's got a lot of money. If you wrote to them, I mean, I have no idea who would have.
Well, I'll take your suggestion. I mean, that's a great suggestion.
Powerful. And, to me, it sounds an incredible story.
It is an incredible story. It is an incredible story. But thank you for that suggestion. That's one of the reasons why I wanted to come, was for help, and that would be a good suggestion. I can comment.
Contraceptive tablets, it carries a clear warning that there are genetic defects for long-term use. There are side effects as well as.
But this isn't written as a side effect.
Yes.
All right, I think we're going to need.
Yes, I'm fine. Okay, let me just unplug here. Thank you very much, all of you.
Thank you. This is yours.
Oh, thanks.
Okay, this should be all set up here. Let's put it on here before we go. It's over here.
Okay.
You want to just run it right off of here?
Yeah, I will. I'm gonna get the thing in there.
Does it not fit?
No, it should. It's just I don't know if I had the right orientation. There we go.
Okay, which one is on? I forgot.
I'm just gonna find it on there now.
Okay, let's close out this one. And this one. There it is. Yeah, you can get it from there, right?
Yeah.
Okay.
All right, so for my presentation, this is going to shift a little bit compared to what you have heard and most of what you probably will hear for the rest of the conference. My work is non-human, focuses on more of an environmental emphasis on coral reefs and the degradation of the ecosystem there. So my interests are really on those chemicals, environmental chemicals, and how they are impacting these non-genomic model organisms. In this case, what I'm looking at here is a sea anemone. I use this as a little model to look at the bigger question regarding coral reefs, because coral reefs around the world are under an unprecedented level of degradation. And we have all heard, about global warming, sea surface temperatures rising, ocean acidification, large, environmental questions that are of global concern. And so, in this particular case, I show you a picture of some coral and a number of the different types of stressors that are known to impact the corals. And you will see here that some of these are natural stressors, but some of these are more anthropogenic, induced by human exposures, just from the way we live our lives and things we don't like bugs when we can, so we'll get rid of the mosquitoes in the Florida Keys, and we'll use pesticides for getting rid of the insects in our house, and herbicides to keep the weeds down as we grow our gardens and agriculture. So you see a variety of different types of stressors, and so the questions that I'm interested in is when you're looking at something like coral, wanting to take and have some idea as to how do we assess their health. Because unlike our human patients, I don't get to ask them a question and they get to respond. So these are some of the common types of assays that can be done with coral. And for those of you who are unfamiliar with the organism, it's actually an animal with a symbiotic algal cell that lives in its tissues. So you're really dealing with a combination of organisms that are there. And one of those, the algal cells, of course, are autotrophic. And so you've got a number of different factors that are associated with it. And here are some of the common ways that coral reef biologists will assess these organisms. They look for growth. They can look for a thing called bleaching. Bleaching is a case where those algal symbionts are expelled from the animal tissues. Now, corals can, when they bleach, they sometimes can recover, but oftentimes bleaching can be a precursor to subsequent death. So that's a common one. There's, of course, a photo to respiration ratio. Since you're working with an autotroph as well as a heterotroph, you can look at the ratios of, those processes that are occurring. Of course, there's also development, larval development, and then recruitment, when you have these planktonic organisms as larvae, and then they have to settle down and metamorphose into the adult forms and then grow and develop into the hard reef structures that we're familiar with. Chlorophyll concentrations within those algal symbionts, and of course, just a general protein production, and physiology, respiration rates, and of course growth is one mechanism. Different kinds of corals have different growth rates. Some of them grow on millimeters per year kind of a rate. Some species can grow up to nearly 10 centimeters per year. So there's some that grow quite rapid, others grow quite slowly. So the question becomes, if you have all these varieties of ways of assessing, how do you determine what's the impact of which stressors having the greatest impact? I show you a picture here of a little coral that is beginning to show signs of bleaching. There's little white spots on this. There's little regions where the symbiotic zooxanthellae, those algal cells, have been expelled. And so I list around here a number of different things that are all known to induce that type of response. Some of these are sedimentation and temperature, those natural kind of stressors. Ultraviolet light, during different seasons of the year, you're going to have higher intensity exposure. They're more directly attributable to the impact of humans. The use of pesticides, the herbicides, heavy metals. These are commonly associated with runoff, wastewater effluent, these types of things. So the question becomes, if you have a whole variety of different types of natural and anthropogenic induce the same response, how do you know which one is the result of this? This particular picture that I took was taken in the U.S. Virgin Islands in February. So in February, ultraviolet light exposure is not at its peak in the northern hemisphere. The temperature was not anywhere near its maximums, so I didn't have to worry about that. The water, the clarity was, sedimentation was quite low. So I can kind of infer that this cannot be the driver, but I really can't eliminate any of these other three. Not at least based upon the time of the year and where I was at. Location was reasonably close to, you know, some places where people were living, so I can't eliminate these. So the question becomes, how do I prioritize this? How do I prioritize which of these factors may be leading the way? So that's where I move into this idea of transcriptional profiling. The kind of work I do is I look at changes that come on the point of when genes are turned on, when the mRNAs are being expressed. This should be the very first step towards a new response, something that is impacting whatever the stressor is. It should be at a change at the level of a gene getting turned on or turned off. An mRNA is getting produced. So the questions that I face, though, is well, there's thousands of genes. How do I find it? Well, it turns out there are some, you know, common trends that I can gain, you know, feedback from the model organisms. And one of the things we know is stressor-induced genes represent a relatively small portion of the genome. In other words, you know, you have thousands of genes, but not all of them are going to necessarily be induced, upregulated, or necessarily downregulated just because you're exposed to a stressor. There are certain kinds of housekeeping genes that every organism is going to have and in utilizing if it's just alive. And so I'm more interested in seeing what are those genes that are likely to be sensitive to these changes in the environment. And the idea is that different combinations of these very specific stressor-related genes can kind of be like a fingerprint. We all have them. They're not exactly all the same, but there are similarities, and there could be certain subsets of genes that may be representative of a particular exposure environment that they may have undergone. So the idea here is that with the transcriptional profiling is if I'm looking for things that are sublethal, obviously we know that many things are toxic and can kill an organism if it's at a high dose. But those low dose, sublethal, maybe short exposure, maybe long exposure, but just very small amounts, should lead to different types of transcriptional profiles for different compounds, classes of chemicals. So as just kind of to help me along the way, I show a slide that's got some relatively well-known, long-time established data just in genomic known models. You know, yeast, roundworms, and fruit flies. You can see that we know how many genes there are in these organisms, these model organisms, and roughly somewhere between 5 to 15% of their genome seems to be regulated by stress responses. So when I want to take a look at a non-model organism, I know I don't need to be looking for a huge number of genes. I need to be looking for a relatively small subset. At least this gives me a guidance for where I want to go. So what I did in this particular study here is I was interested in the idea of kind of endocrine disruption. And so I chose estradiol to look at with a species of anemone. I know that there was evidence of estradiol that was present in the water columns in corals when they release, in corals when there's mass spawning, the gametes are shed into the water column, and on the Great Barrier Reef, these spawning slicks can literally be kilometers in length. And so the water testing has detected the presence of the chemical there. But when it comes to cnidarians, there's not much that's been well characterized regarding hormonal signaling and any kind of bioregulatory pathways. In this case, cnidarians don't have estrogen receptors. So what's going on if there's some kind of estradiol, what is going to be the impact? It's got to be, due to the hydrophobic nature of the compound, passing through the membranes and having some kind of interactions, nuclear receptor or nuclear binding of some sort. So I'm using the small anemone. It doesn't produce the hard calcium carbonate like the corals do, but just a sea anemone called exaptasia. And it's easy to keep in a lab. You can use it in small little bowls. You can keep it in a small aquarium. In the aquarium trade, all the people who keep nice saltwater tanks, this is their pest. They don't want this in there. They're always trying to get rid of these. Anyhow, there's been kind of a movement to try to make exaptasia one of those model organisms to study. And as I point out, there are no estrogen receptors in cnidarians. So I said, I want to see what's happening here. And so the way I went after this is I exposed them to a 20 parts per billion treatment. In ecotoxicology, ppb kind of is the units operated with typically. If you're not familiar with that, these are going to be low nanomolar, possibly even down to picomolar concentrations. And I chose 20 ppb, not because this is an environmentally relevant concentration, but I wanted to kind of expose them to a level that was enough to where I can generate a response that I should be able to see something. And then I did it for just 4 hours. And then I used a protocol called representational difference analysis. This is a form of a subtractive hybridization, but I think there's some advantages that I'll point out in just a minute. And then from there, I can generate fragments of, I take the messenger RNAs, reverse transcribe them, I have cDNAs, and then I can take with the RDA, it'll chop it up into smaller fragments, and I can get some of these fragments of these responsive genes, and they become the genes of interest. It gives me sequence information that I can use, and then I can quantify the abundance by doing some quantitative real-time PCR. So, if you're unfamiliar with this, just briefly, the technique, like I said, it's a form of a subtractive hybridization. There are commercially available kits for this. I would suggest, though, that the commercially available kits don't provide the sensitivity that this does. The reason why I say that is that this protocol helps to, I think, in a greater way, selectively enrich for those rare transcripts. This is kind of a PCR-based process. If you're unfamiliar with these protocols at all, this is where you take pools of mRNAs, reverse transcribe them into cDNAs, and then eventually in the whole process, you will take and combine those mRNAs that were originally expressed by those exposed and those that were not, and you bring them together. And through PCR, the only ones that will actually amplify will be the ones that are uniquely associated with one population. I'd say the majority. That's how you enrich to find these rare transcripts. And so what you can find is you can simultaneously find genes that are either turned on or upregulated, or you can find genes that are turned off or downregulated. So you start to screen through these, and you end up with a series of small fragments of gene sequence, and you can then use those as the basis to either build or assemble or include on microarray platforms, or you can do qPCR assays. So this is just kind of a quick overview. You do a series of hybridizations, and they selectively will enrich. And so in the first round here, you get what looks like a pool. I clone the pool, and once you've cloned the pool and separate them out, you see you have various individual sized fragments that occur. You see some fragments here are all the same length. Those may be replicates of the same thing, but they may be bands of different sequences that are co-migrating at the same length. Yeah. So and you can see then that as you go through this first rounds, they look very much blurry, smeared, but by the second round, they become more focused. And because of the proportions of the two populations as you mix them, the rare transcripts start to show up in the more subsequent rounds of the hybridization that occurs. So I'm just going to jump straight on into some of the data that I was able to find. Once I had cloned these sequences, I analyzed the sequences with bioinformatics. There were certain sequences that popped out at me that I thought, huh. Now I knew I had used estradiol in my exposure, and one of the ones that came up was the 17-beta hydroxysterol dehydrogenase enzyme that's in the pathway. So I said, I must be on the right path at least. Now, the real focus, what's this doing in the cnidarians other than modifying a sterol, that's all I can tell you at this point. But I thought, this is at least a clue. I must be working along, I must be isolating something of significance here because I can directly link this enzyme to the environmental exposure that I just did. And then I started seeing some other things, and I didn't necessarily know always what was happening. This showed up a lot. I got this Niemann-Pick C. I thought, I don't know what this is. So I do a blast search, and I start reading, and this is a molecule that is involved with sterol transport, cholesterol transport specifically. And so this was showing up, and then I got C3, an immune response. And then I also had a histone demethylase response. So I had one other one that popped up, which was an interesting one. I had this enhancer binding protein. The reason why this one was interesting to me was because I did a 2012 study where I looked at this using the same anemones. I exposed them to a pesticide. And in that work, I saw this very same gene. So for the purpose of just this meeting here, I went ahead and pulled out of some of that data from that 2012 pub, and I put it on here too. And I had to transform it because it wasn't done with qPCR, but I normalized and transformed that 2012 data with the organochlorine stuff to show a comparison. So the same gene had a very different response. The Lindane work had a series of different concentrations of the organochlorine, and I compared that to the estradiol. And so the blue, if you'll notice the blue, you can see it's the same gene is responding very differently with these different chemicals. Okay? All right, so I took a little bit closer look. What are these different chemicals, these 5 genes, what do we see? And well, this enhancer binding protein, this is a transcription factor. And some of the characteristics that we see in other organisms, well, actually I found some studies that show this is involved with nerve cell development. And so, getting a clue here, we're talking with estrogen that we think of associated with development. Now I'm seeing some nerve cell development as it relates to this one. It's a regulatory enzyme associated with antioxidants, so there must be kind of an oxidative stress component to this. Xenobiotic transformations seem to be some linkages also with, so this particular gene now I'm starting to see, okay, estradiol, and I'm seeing a transcription factor that has these other characteristics. And then, of course, activates other responsive elements related to oxidative stress. So now my signs are saying, okay, oxidative stress, estradiol. Those are some of the things that I'm seeing here with these exposures. The hydroxysteroid, once again, I know there's a little bit about this. I mean, I'm not a detailed and extensive endocrinologist in my work, so I'm going to learn a lot more about what is going to be the impact of this and how does this enzyme modify these large sterol structures. The Niemann-Pick, once again, it's a critical mediator involved with cholesterol absorption, okay? So we see as the transporter plays a role. Some other work with in mice, we find out that when this is knocked out, you have a reduction of estradiol in cells. So why is this upregulated in this particular case? Well, we know I've dosed them with a higher level, so there must be a function that's going on there. And in cnidarians in particular, the presence of this enzyme or this transporter seems to be associated with the interactions between the algal cells and that animal host. The histone demethylase is one that really jumped out at me. And that's why, because there's some studies to indicate this is kind of an estrogen-induced growth. So I'm getting clues that, once again, development and growth is involved here. This methylase is also representative of epigenetic changes. Epigenetic changes can either be the DNA methylation that we've heard about this morning. It can be changes in the methylation pattern of the histones, which is what I think I'm showing you here. And then there will also be epigenetic changes regarding microRNAs. So what you can kind of see here is the epigenetic changes. If the genome of the organism, the DNA is like the hardware, it's these epigenetic changes that are oftentimes referred to as kind of the software that runs the system. And so this tends to, once again, gives me a clue that says maybe these are ways that we're trying to, this environment that I'm exposing, that I'm manipulating, is starting to potentially has an impact on programming that genome. The C3, the interesting thing about this is cnidarians don't have an adaptive immune system, but the innate immune system has C3 components of it as well. So I'm seeing that. So the environment exposure seems to now also be triggering some type of a immune response. I think that seems to be a repetitive theme I see in my work, is that these chemicals, anthropogenic chemicals, stimulate immune types, innate immune responses in these aquatic organisms. We also know that the C3 in cnidarians does seem to have a link to injuries. There's some other studies suggesting that. And so this may be one of those targeted molecules or targeted gene transcripts that may be a link between immunity as well as development. All right, so what I kind of have with these 5 genes that I've showed you is that we can transport sterols. We tend to modify sterols. We're altering the genome because of exposure. There's an oxidative stress component, and then there seems to be an immune thing. So we've got a combination of different processes, and it's this combination I'm going to propose we may start to get a picture, a profile, that when an organism, in this case these anemones, are exposed to the estrogen, it tends to create this kind of a profile which may lead to, figuring out, can a suite of genes potentially be an identifiable marker to represent exposure to a particular class of chemicals. So the next step that I want to do with this work is I'm going to go back now and expose the anemones to a whole series of concentrations. I know that 20 ppb is above an environmental relevant concentration. Now I'm just going to take and work my way back down and do a whole series, and I want to take and see if I take these 5 genes and start to look at different concentrations, at what point do they peak? What are the dose responses going to look like? Okay. Also, I think because of this epigenetic component, I want to see what happens, how long will this be sustained, and what does it look like if the chemicals have been removed? I'd like to see what that does. And then I'm also going to start doing some stuff between, chemical, I've got some anemones. I did some of this work out here on the West Coast as well as here with the Aptasia on the East Coast. I'm going to start making some comparisons between the species. So to wrap this up, I can quantify some very detectable changes using this approach. There does seem to be some evidence of some estrogen responsive genes that may represent as kind of biomarkers of an exposure. And as I do some of these further screens, I may be able to refine and get a little more clarity as to how well these may be representative of that. The representational difference analysis, I think the strength there is, I work at an undergraduate institution, and so my budget is small, And I don't have the luxury of, I would like to be able to do whole transcriptome analysis, which is becoming quite, functionally, attainable now, but it's still quite expensive, and it generates an enormous amount of data. And a lot of it has to still be filtered. So instead of starting big and focusing down, I can start small and kind of come up from the bottom. And so I get a pretty good bang for my buck starting with this approach and filtering through. I don't get all of the responsive genes, but I will get a subset that I think are representative or potential candidates for further investigation. And then, of course, the question becomes is if there's an epigenetic component, as we've heard before, if this can be kind of followed up and established, then we could be looking at brief exposures may have much longer impacts as a result of their exposures. And then, of course, where this all really shakes down is because there's a lot of evidence to show human pharmaceuticals are being released in treated sewage effluent. You can find, you do the water quality out there at the effluent in the oceans, outfalls, and you can detect estrogen. You can detect beta blockers. You can detect analgesics. And so I'm, when I'm looking at these things as it relates to cnidarians, this is a wide open field. And so that's why I'm moving in that direction. I'll stop with that. You probably don't know where Berry College is or what this is, but I put this slide up because it's true. It is the largest campus, and if you want to have an idea of how big this is, it's basically the size of the city of San Francisco. So it is the world's largest campus. Now, it obviously doesn't fill with that many people. It's a small subset where the physical buildings are, but it's 27,000 acres. So,
Just a quick question. Can bleaching be induced by oxidative stress then?
yes, actually, well, yes, and oxidative stress is well characterized.
So then there's a link to the talk by Dr. Anderson earlier today when you mentioned that estrogens
Yes.
cause oxidative stress.
Yes, I think that's clear. There was another slide I could have put up, and I was trying to think, I just can't quite make it simple enough yet, but I could. There's an oxidative stress component, there's an immune component, there's a developmental component, there's transcriptional reorganization or chromosomal reorganization, and there's another one I had in there as well as the steroid sterile transport. There's a whole number of components, so it's kind of finding the balance between these. Yeah.
What is the toxicological hazards of estradiol on the coral reef, or you call it cnidarian?
Cnidarian.
Yes. What is the toxicological hazards of estradiol on it?
Well, at this point, I don't know other than it would be those that are in close proximity to sewage outfalls. Mm-hmm. I mean, in the Florida Keys, there's no sewage treatment. It's all septic tank, mm-hmm. and they're in limestone, so it's very porous, and there's a lot of indication that there's leaching that's occurring. But of course, in any major city, there's some of the work that I did off of Miami, big old outfalls are sitting right there, and it's just getting pumped out. So, there is a potential for some kind of an impact in some kind of a localized area.
Okay, thank you.
I know we're over. Maybe we can talk.
We're just going to say about the estrogen effect. I mean, in Norway, which we've just been mentioning, there's an enormous problem with the estrogens and affecting the reproductive system. So why should this be any different? It is a really big issue, estrogen effects.
Yeah.
And it's already, from the women for which nothing's been done about it. Yeah, I mean, it's been a problem for years, and nobody sort of.
Well, I think that but I think the distinctive element that's on this work I'm showing though is the fact that it's because there are no receptors here. So we're looking at simply a hydrophobic chemical and the passage.
If
But
there
I
are
agree.
no receptors, there's this other route, which
Yes.
is really,
Yes.
really, and people have ignored it.
Yeah.
But it is oxidative. I mean,
Yeah.
end up with the hydroquinones and things like this,
Yeah.
which are incredibly potent.
Yeah. And so in that sense, that's where I think this work kind of complements that. But I know we need to wrap this up, but maybe we can speak later for other further questions.
And the thing is that in some countries, they have already started to grow this cemetery,
Yeah.
with resistant active.
Yeah.
Just for avoiding this problem.
Yeah.
Okay, I'm gonna take 2 more minutes and do this, and then
You should be fine. You don't- Just leave it there and we'll get it out for you, so you should be minute. I don't want to ruin your data.
Okay, so this is kind of thrown into the schedule here at an unusual space, but I do want to go ahead and before we go to lunch, first of all, does somebody have a camera to take this group picture? Is there somebody who's going to take care of that? It's written in my schedule, group photo. You have one? You want to do it with that? Okay, so maybe would you like us all to gather up here afterwards? Okay, so we'll do that. But before we do that, I have some nice certificates here for the organizers, the organization committee. So I have 4 here, and I apologize. I'm not really good with names. So if I could have Ying Peng. From Sun Yat-sen University in China, is that close enough? And Friday Uba, is that close? From the University of Calabar, Nigeria, T.G. Borovskaya from the Goldberg Research Institute of Pharmacology, Russian Federation, and Marcelo Laramendi from, let's see, National University of La Plata, Argentina. If you guys will all come up, we'd like to present you with these certificates to thank you for your help in organizing this. So does somebody want to come up and help me? Can you come up and help me again? Okay, so let's see. Sal, here's one for you. Okay. Get back to him. We'll try to make this look formal, right?
Let's be formal.
Thank you.
The TG. This is TG.
Right?
That's you.
And this is, you won't have any trouble, sorry. Very well. And then,
Okay, now we all get to be in a picture, so let's all come up to the front where he can see us from that camera, and then we can go have lunch. I think. That's what the paper says.
Testing panel. 1, 2, 3, 4, 5, 6, 7, 8, 9 on the panel mic. Panel mic 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5. Are there only 2 people sitting up here at the panel? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, or do you have 4? Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 at the panel mic. Sir, I will help you with the laptop in just a second as soon as I get done with this. So I can help you out. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5.
Talk while you're helping.
Exactly. Thank you. I'm going to help you. We're going to load this right up for you, and then you'll be ready to go. All right, we're talking, we're loading. I'm doing a mic check. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12.
talk today will be primarily.
No, that's microphone turned on.
My talk today will be primarily about if Sonia very little is known. Which and metformin are being increasingly used to treat gestational diabetes because of their ease of administration and efficacy comparable to insulin, but at much lower cost. Then the thing is really about when the pregnant women with gestational diabetes are treated with globulide, their fetus are also exposed to the drug de facto. And in fact, clinical studies have shown globulide can actually cross the placenta barrier to the fetus at really some level. For example, the umbilical cord blood concentration of globulide were just slightly lower, about 70% of the maternal plasma concentrations. So which is very substantial. So the question is really now how safe this medication is to the developing fetus. To address this concern, there have been a number of clinical studies to compare neonatal outcomes in the offspring of women with gestational diabetes receiving globulide or insulin. The data were actually quite contradictory. There have been clinical studies showing basically no significant differences in the percentage of infants who were larger for gestational age, fetal microsomia, neonatal hypoglycemia, neonatal intensive care unit admission, and or fetal anomalies. So basically, this clinical study showed globulide was actually as safe as insulin. But then there also have been studies reporting a trend towards higher incidence of infants with microsomia or larger for gestational age, and a significantly higher rates of neonatal hypoglycemia in the globuli group compared to the insulin group. So basically, those studies show contradicting results in terms of fetal safety of globuli, particularly both long-term and short-term. Particularly, we don't know anything about long-term safety of globuli because none of those clinical studies really actually monitored those babies for a long time after birth. As one of the efforts to address this concern, because I'm a pharmaceutical scientist, I'm really interested in understanding the mechanism that controls the the quantity of globuli exposed to the fetus during pregnancy. So hopefully understanding that would allow us to figure out, is there any way to predict, to control, or to predict, to prevent fetal exposure to globuli, based upon mechanistic understanding of globuli disposition in the fetal placenta unit. So that is the topic of my presentation today. Okay, a little bit about the background, about the structure of human placenta barrier. As you can see here, placenta barrier is composed of primarily a layer of sensitive trophoplast, which is shown here as a tree-like structure that separates the maternal circulation, the pink area, and the fetal compartment. If you look at the cross-section of the sensitive trophoplast, the brush border or apical membrane faces maternal circulation. The basal membrane faces fetal compartment. And there are a bunch of different transporters expressed on both sides of the cell. Notably, the two ATP binding cassette efflux transporters, PGP, BCRP, are highly expressed on the apical membrane. Because of their substantial and strategic localization on the apical membrane, they are presumed to protect the fetus by limiting fetal exposure of medications, xenobiotics, by pumping them from the fetal compartment back to the maternal circulation. And we have previously shown that globuli is actually a very good substrate for BCRP. And therefore, we have hypothesized that BCRP is important in limiting fetal exposure to globuli. To test this hypothesis, we did a transplacenta PK study in pregnant mouse model because such a transplacenta PK study cannot be done in humans. Okay, this is what we did. We dosed the globuli to wild type and the BCRP knockout pregnant mice by retroorbital injection, which mimics IV dosing. And then we collected the maternal blood and the fetus at various time points after drug administration, and then quantified the concentration, globuli concentration in the maternal plasma and the fetus by a validated mass spec assay. So if BCRP does limit fetal exposure to globuli, we would expect that the BCRP knockout fetus will have a higher globuli concentration and exposure than the wild type fetus. So that is the data. So it turns out that the knockout of BCRP actually did not significantly affect the maternal plasma concentration profile, as you can see here. However, the knockout of BCRP increased the fetal concentration of globuli, but especially after 30 minutes. Then we calculated AUC value. AUC is the area under concentration time profile, which is a PK parameter that is used as a measure of exposure of medication in the body. Okay, the knockout of BCRP only slightly decreased the maternal plasma AUC. However, the knockout of BCRP increased fetal AUC about twofold. as you can see here. Then the ratio, the fetal to maternal plasma AUC ratio was also increased more than twofold in the BCRP knockout mice compared to wild-type mice. So the conclusion is BCRP1, the myelin homologue of human BCRP in the mouse placenta, limits fetal exposure to globuli. The human BCRP may play a similar role. This is a study showing BCRP is important in limiting fetal exposure. Nonetheless, we know from the clinical study, we know BCRPs can still cross the placenta barrier by passive division, despite the role of the function of role of the BCRP in limiting fetal exposure to globuli. So then the question is, what happens once the globuli gets to the fetus? We know in human adults, we know that BCRP is extensively metabolized in adult liver by cytochroma P450 enzymes such as CYP3A4, 2C9, 2C19, and therefore we hypothesize that globuli also undergoes metabolism in the fetus by CYP enzyme expressed in fetal livers. So to test this hypothesis, we did a microsome incubation study with both the human adult liver microsome and human fetal liver microsome. And this is the data. Interestingly, with the human adult liver microsome, you could detect actually all the metabolites, six metabolites. With N5 is a major one. They all can be detected at a substantial level. However, with the human fetal liver microsome incubation, only the N5 can be quantified. Other metabolites could be detected, but at really very, very low level compared to human adult liver microsome incubation. So these are significant differences, difference in metabolic profile between the human adult liver and human fetal liver microsome. And we know the human fetal liver, in human fetal liver, the major CYP enzyme is really the CYP3A7. In contrast, in adult livers, the major CYP3A isoform are actually the CYP3A4 and the CYP3A5. So we thought the differences in metabolic profile is maybe due to the differences in metabolic profile of those individual CYP3A isoforms. And therefore, we did a similar incubation study with a recombinant individual CYP enzyme. So this is the data as shown here. As expected, the CYP3A4 and the CYP3A5 generated a metabolic profile that is very similar to human adult liver microsome. However, CYP3A7 generated a metabolic profile that is similar to a human fetal liver microsome. And therefore, such analysis of metabolic profile suggests that CYP3A is likely the major enzyme responsible for globular metabolism in fetal livers by human fetal, in the fetus by human fetal livers. And N5 is a major primary metabolite in the fetus. And then we went ahead to determine the kinetics. So the kinetics basically allow us to determine the Km Vmax and the intrinsic clearance. Intrinsic clearance is a Vmax divided by Km. So intrinsic clearance is an indication of the intrinsic capability of enzyme in catalyze a specific metabolic reaction. It turns out that CYP3A is 3 times more efficient in catalyzing the globular depletion than CYP3A7. Then the question is, responsible for globular catalytic efficiency. Because the contribution of enzyme to the metabolism of a medication in the organ depends on not only the intrinsic clearance, but also the expression level. So therefore, we went ahead to quantify the protein expression of the three CYP3A isophones in individual human fetal livers. So we did that using a highly sensitive mass spec assay, and it turns out that CYP3A7 is actually the only enzyme that can be quantified. All other enzymes, including CYP3A4, cannot be quantified or cannot be detected, or could be detected but but could not be quantified because of the lower level of expression. Interestingly, there's a strong correlation between the CYP3A7 protein content with the product formation activity from CYP3A7 prober substrate. That is a DHEA, as you can see here. So there's a strong correlation between the 16-alpha-OH DHEA and the CYP3A protein expression. And there's also a strong correlation between the globuli depression activity and the CYP3A protein expression in 16 individual human fetal livers. Likewise, there's a strong correlation between the globuli depression activity and the 16-alpha-OH DHEA formation. So those are the data really support the conclusion that CYP3A could be the major enzyme for globuli metabolism in human fetal livers. So then we did an additional study using another substrate, midazolam. We know midazolam is actually a proper substrate for CYP3A4, which characterizes a hydroxylation of midazolam, preferentially at 1' position over 4' position. And because we know now CYP3A4 expression level was very low in human fetal liver. So the ratio, the metabolite 1' OH to 4' OH ratio, metabolite ratio would be expected to be much lower than 1 for majority of the human fetal livers. So that is exactly what we found. And again, interestingly, we also found that there is a strong correlation between the CYP3A protein content and the 4' OH midazolam formation, as shown here. Here, this one, yeah. So suggesting that the CYP3A7 preferentially catalyzes the hydroxylation of midazolam at the 4' position. And then, as expected, there was no correlation between the globuli, or there was a strong correlation between globuli depression activity and 4-OH midazolam formation, and there was no correlation between the globuli depression activity and the 1' OH midazolam formation. So those are the additional evidence further supports that CYP3A7 is a major enzyme for globuli metabolism in human fetal livers. Okay, now conclusion, I would say globuli can cross placenta barrier by passive diffusion to a certain extent. Even though we know BCRP plays an important role in limiting fetal exposure to globuli. And then once in the fetus, globuli is metabolized by CYP3A7 in human fetal livers. So in the future, I think it is important to test if the major metabolite, N5, is pharmacologically active, because we know other metabolites, N1 and N2B, are pharmacologically active. And then the results imply that the alterations in BCRP and the CYP3A7 expression or activity in the fetal placenta unit can affect fetal exposure to globuli and its metabolite, and ultimately the fetal safety of the drug. You know, at the beginning I said that there are a lot of variation or contradicting results in terms of fetal safety of globuli. In the clinical study, I think at least part of the reason could be due to the variation in PCRP or CYP3A7 expression or activity, that the contributing factors could be genetic polymorphism or the variation in the exposure to medications or xenobiotics. All those factors can change expression or activity of transporters and enzymes. And I think it is important to test this possibility in future studies. And those studies are the essential for PBPK modeling prediction of fetal exposure to globuli. Okay, I appreciate, I would greatly thank my previous current postdoctors, graduate students from my lab. Also should never forget the taxpayers' money. Let's support our research. Okay, thank you. Any question? Welcome. Yeah.
We have time for maybe one question. Is there one question?
So what happens to the fetal metabolite? Does it go back into the mother? Does it diffuse back there? Or because how does the fetus get rid of the metabolite?
So you are asking what happens once?
Yeah, so the CYP3A7 metabolizes.
Yeah.
Then what happens to the metabolite? Does it go back into the mother and the mother excretes it?
Some of them may remain in the fetal compartment. I think a majority may remain in the fetal compartment, yeah. And some may be transported back to the maternal circulation. But we don't really know anything about that. What is what is the disposition mechanism of the metabolite? Yeah, we don't. No study has been done to that. You know, but I believe majority of the metabolite may stay in the fetal compartment, and that's why it is important to know if it is pharmacologically active. Yeah.
All right, let's go ahead and try to move forward. We have a little change to the sequence of presenters. Mary Staley is going to change places with Kristen Comfort. And so Dr. Comfort's going to present her research now.
All right, thank you guys very much. I'm tasked with the challenge, I guess, of keeping you awake after lunch. So I will do my best to kind of move through this, get us closer to back on track. This doesn't really fit in this session because family emergency and they move early, but what my talk is on is the need for nanomaterial evaluations to take place in a physiologically relevant environment, and then being able to connect environmental variables to nanoparticle behavior and resultant toxicity. So recent studies have identified that nanotoxicology is really dependent on what is referred to as the nanobio interface. And this is the dynamic, ever-changing interface and interactions between the nanoparticles that's driven by kinetics, thermodynamics, and transport phenomenon. Now we know that this nanobio interface is influenced by factors on both the material side as well as the bio side. So how does the shape, how does the surface chemistry of the nano change the interactions, as well as what cell type we have, what are the expressed surface receptors. So these two factors combined give us our nanobio interface, which determines the degree of cellular association and internalization, as well as nanomaterial behavior, which is what this talk is going to focus on, is then those two combined we know lead to our observed cellular consequences, whether it's toxic, stress, immune response, etc. All right, so the motivation for the work that I do is that up until now there have been tremendous advances in the nano side. So the characterization, the synthesis, the dosimetry have always been a major focus of nanotoxicology, but what has always been lacking is that same progress on the bio side. So if we get that parallel progress, then we will leaps and bounds be able to better understand nanotoxicological effects. So the long-term goals of what my group does is to generate in vitro models that can closer mimic an in vivo system, as well as to improve predictive capabilities and design potentially high throughput analyses. So I'm going to take a step back. I know nano is not something a lot of you are familiar with, so I'm going way back to kind of get an encompass for everyone. So let's compare the in vitro and vivo. So these are both being used for nanotox, and they both have advantages and disadvantages. On the in vitro side, it's a simple model. It's quick, it's cheap. We can go very deep into the mechanistics, and we can say specifically, I want to look at this model, this type. The negatives obviously are you can't extrapolate that to humans. It's way too far removed. On the flip side, we have the in vivo models, the animal models. Those are phenomenal for advanced physiological responses. We get the whole picture. We get the big picture of what's going on. Disadvantages, as you guys are aware, the ethical concerns, the cost, the time constraints. What you might not be aware of is the dosimetry. Nanoparticle dosimetry is a huge hot topic right now, and we have no way of really correlating in vitro and vivo dosimetry very well. So these differences have led to poor correlation between in vitro and in vivo nanotoxicological. Something that will be cytotoxic and in vitro, you throw it into an in vivo model, nothing happens. So we need some way to bridge this in vitro and vivo gap and find a way to analyze nanoparticle effects in an efficient manner, but that is also accurate. So that's the focus of what I do. So what I'm trying to do is take in vivo and shrink it down. So let's start with if we had a piece of liver. What are some of the attributes of a chunk of your liver? First of all, it's three-dimensional. Secondly, it's comprised of multiple cell lines. If we're talking about liver, it has hepatocytes. It has endothelial cells, it has epithelial cells, it has Kupffer cells, which are the immune cells in there. So it's not a single monoculture. It also has physiological fluid, whether it be interstitial fluid or if you're in the bile secretion areas, bile, something in there, it's not cell culture media. It is a biological fluid. And lastly, everything in our body is either directly or indirectly connected to the cardiovascular system. So we are not static beings. Even if we are to sit perfectly still, our bodies are still moving. However, traditional cell culture is static, so we're overlooking that major dynamic movement. So the primary goal is to take traditional in vitro, which is 2D monoculture in cell culture media, static, and transform it into something that's more representative of our complex model, with the long-term goal of hopefully we will be able to increase predictive correlations between those two guys. All right, so the work I'm going to present is one study. I've done a handful of these, but this is a master's student of mine who just graduated, so this was actually her thesis work. So what Emily did was look at an alveolar region. This is of particular interest because inhalation is one of the primary forms of nanoparticle exposure. For instance, aluminum nanoparticles are in gasoline. Gasoline gets kicked out of cars, we breathe it in, that's how we get inhalation exposure. So our model contained human alveolar epithelial cell lines. It also included an artificial alveolar fluid as well as dynamic movement. We used 60 nanometer tannic acid coated particles and looked specifically at how this altered in vitro system affected their characterization and the nanobio interface. Our dynamic flow was set up through the use of a multi-channel variable flow peristaltic pump. We connected each of the channels exclusively to a single well of a 24 well plate, which gave us unilateral flow across our cell system. And we chose our target volumetric flow rate very carefully to give us velocities within the tubing of 0.2 centimeters per second, which is the capillary flow rate, which then meant the velocity across the cells was orders of magnitude lower, about 0.003, which is representative of diffusion. So this is actually a really cool system because it simultaneously captures the cardiovascular systems represented by the tubing and then the indirect effects of what's going on in the cell. So since the lungs are not part of the cardiovascular system, that doesn't mean they're not affected. It just means your fluid movement is slower. All right, so the first thing we did was verify that the cells were viable. I'm not showing it here, but we also did run a handful of standard viability assessments to make sure our cells were not dying either under dynamic flow or with artificial fluid. And what you see is significant change in cell morphology. So figure A is the A549s in static media. They're globular, they're round. It's what we're used to seeing, they're happy looking cells. When we move and keep static but replace the media with our artificial alveolar fluid, they're still globular for the most part, but what we see is this unique curvature of the membranes. You no longer have kind of the wavy, it's a very defined membrane line. And when I dug into the literature, we actually saw that this is common in vivo alveolus as well, and this is brought about by the high tension of alveolar fluid, and it impacts the membrane. So we're seeing this in vitro as well as what has been reported in vivo. When we add dynamic flow to that mixture, for C, which is media, you see an elongation in the directionality of flow, so our flow is moving across horizontally the screen. And in D, which is both alveolar and dynamic, what we see is both that elongation as well as that curvature again. So in addition to the curvature being seen in vivo, we know that shear stress applied across cells does cause them to elongate. This is how you get polarized cells. So these two alterations to our system did allow us to capture in vitro what's more representative of what's actually going on in an in vivo system. All right, so here are the particles we were dealing with. They were 65, they were actually, well, they were supposed to be 60-ish. You never know exactly until you characterize them because each batch is different, but they were 65 nanometers, tannic acid coated. When you put them in water, they agglomerated to 75. Keep that 75 number in mind. Zeta potential, so their surface charge was about -30, and this is brought on by the surface coating of the tannic acid. And they did have a slight ionic dissolution. I did ionic dissolution with the gold, even though that's not a major factor with gold, but with a lot of the other metals, silver, copper, titaniums, the dissolution has been shown to cause the cytotoxicity. So I did dissolution of the gold just for comparison. All right, so what happens to these particles acellularly to start with when we put them into this microenvironment, artificial fluid and dynamic flow? Well, first, if we look at the spectral signature, the gold has a unique peak at about 520 nanometers. When we go from water to media, that peak is slightly changed, a little bit of a right shift, but essentially they're the same. However, when we resuspend these in alveolar fluid, our peak bottoms out. You get peak broadening, loss of spectral intensity, and you also get the addition of a new peak at about 700. So what this means is we have a lot of agglomeration going on. We have particles falling out of solution, and we are losing stability of our particles once they're redistributed. So to confirm that, I next ran Emily, my student, ran DLS, so dynamic light scattering, to get agglomerate sizes. So in water, I said they were 75 nanometers. In media, it's about 100. You expect that, that's the formation of a protein corona. No big change there, but once we put this into our alveolar fluid, the agglomerate size bumps up to 250. So over twice of what we're seeing before. Just by putting these particles in the fluid, they clump together, they agglomerate, they form this big mass instead of little particles, and then they start to sediment out, which is what's causing that change in our spectral profile. If we look at the dissolution, in media we see a spike. Versus water, this is what happens with the ions in there. But then when we switch over to artificial alveolar fluid, we see a drop in that ionic dissolution. Ionic dissolution is a function of size, so the smaller size you have, the higher rate of ion production. Well, once we go in alveolar fluid, they're not little individual particles, it's a big clump. So that big clump is going to have a smaller rate of dissolution, and we were able to capture that here. All right, so once we know what's going on with the particles themselves, we next took this into our cell system and added the A549 epithelials and looked at their deposition. We want to see how much or what fraction of these particles were either bound to the surface or internalized. And as you can see, if we look first at static, so no fluid movement, we had minimal deposition in media, but as soon as we swapped out media for AAF, that deposition more than doubled. Again, this is because of that large agglomeration. So these particles, as we showed, they're not stable. They're not diffusing, they're sedimenting, they're falling out of solution. So essentially in alveolar fluid, they clump together and they fall down onto the cells. So the cells are seeing more particles. When we add dynamic flow to that, nothing changes with the alveolar fluid because sedimentation is the primary transport mechanism at this point. We do see a drop though with media. So in media, we're not having that agglomeration. So all of a sudden when you have fluid movement that way across, it's preventing particles from diffusing downwards and reaching the cells. All right, so to look at what's happening, are these particles getting into cells? We next looked at TEM images. So A again is static media. So I'm going to start with media. So if you look at A and C, that's media that's static and dynamic respectively. And we see small agglomerates inside the cells, not a massive change, pretty much in agreement with what we saw before. There's small pockets, they're getting in there. B and D is our alveolar fluid, and what we see in B is static, and we see some cells inside the cells, or I'm sorry, some particles inside the cells, and as you can see in the blown up version, it's a much larger agglomerate. There's a lot more particles clumped in there together. But there's also a number of particle clumps bound to the membrane but not internalized, and that's indicated by the arrows in the figure. When we look at artificial fluid with dynamic flow, which is D, everything is on the outside membrane. Nothing is actually internalized. They're tightly coupled and bound to the membrane, which is why we're getting deposition, but the cells are not actually internalizing the particles. All right, so to look at this in another way, we did a combination of fluorescence and dark field microscopy. So in this, our actin's red, the nuclei are blue, and the particles with dark field appear as white. So if we're looking again at that left-hand column, it is our media. We see significantly fewer particles than with the alveolar fluid, which is on the right, and that is in agreement with our deposition data. It's in agreement with the TEM data. But what we also can see is even following exposure, those morphological alterations were retained. We still see the elongation with the flow, and we still see the curvature of the membranes with the alveolar fluid. So this is what we think is leading to that change in D in particular, because the cell membrane is stretched out, it's curved, that the focus of the energy source is on maintaining this morphology and not internalization, which is energy dependent. So, like I said, this is just one study. I've done a couple others and getting some very promising results and preliminary data to go along with this. Takeaway message that I wanted to get across today is really it is possible to modify and alter an in vitro system to make it behave more closely to an in vivo, to help us bridge that in vitro in vivo gap. In this study, we looked at dynamic fluids and dynamic flow and artificial fluids simultaneously, and we're able to successfully culture the cells and get them to behave what's more closer to in vivo. And in these conditions, the nanoparticle characteristics as well as their behavior were significantly altered, and there's a plethora of data out there and a plethora of studies that show that this behavior and these characterizations is what leads to the nanotoxicological effects. So therefore, by continuing to play with this system, modify the in vitro behavior, this will allow us to identify novel responses that were previously unobtainable through these systems, and goes back to the original goal of making these two more align and providing a rapid and reliable way of assessing nanoparticle toxicology. So thank you, and thank you again for switching.
All right, we have time for just one or two questions. Yeah.
Did you stabilize the nanomaterial? Did you stabilize it in cases?
It's tannic acid coated, so they are stable, yes.
They are, okay. I didn't say, thank you.
There's a question back there.
Oh, sorry.
Next row. Oh.
Did you consider co-cultivation of your epithelial cells with endothelial cells in a more sophisticated 3D model?
Not in this study, but in a different model, I actually have a lung co-culture that's the epithelials with macrophages to get the immune response. And we see very similar, it's actually very interesting, especially with the morphology, because the epithelials will still spread out, but the macs don't. They stay circular. So it allows us to see both visually, especially with the microscopy, that the macs do in fact preferentially engulf all the nanomaterials, because you'll see all the cells spread out and then the macs scattered, and that's where all the white is. So there's a different study that I'm wrapping up with a different student where we did do a co-culture, but not epithelial and endothelial simultaneously, because they would experience different shear rates. So I'm not sure how well they would go together.
Thank you, and maybe you want to have a look at the poster I presented at a conference earlier last week.
All right, thank you.
All right, let's go ahead and move.
cup.
Okay. all
Yep.
right.
Do you know what happens to the nanoparticles after they're deposited? Like, do they get released?
That, they go through a cycle of things, and it depends, again, on a lot of stuff, the coatings, the sizes. I didn't track them in here. They can be degraded, they can be re-released. And again, that's one of the major disadvantages with in vitro. In vivo, we can follow them and say, we know they end up in the liver, the spleen, the kidneys, but here they kind of capture and disintegrate or capture and release.
Okay, thank you.
Thank you.
Dr. Kumar, you're. We're moving on. Okay. All right. So our next presenter, the researcher is going to be presented by somebody different than who conducted the research. Sergey Alexeyev is going to actually make the presentation on behalf of Kiji Boriskaya. Can we have that mic turned back up?
Huh?
Get them to turn that mic up for you. There you go.
Before I start, I have to note that there is a mistake in the name of the institution we work. In the program and in the proceedings, on the page 43, I think, there is a wrong name. It's written that we work at the Cancer Institute, but this is the right name of the institution, Research Institute for Pharmacology and Regenerative Medicine. Before I stay on the subject of the report, I would like to draw your attention to one fact. The study of reproductive toxicity is an obligatory stage of preclinical toxicology studies of new drugs. The only exception is for anti-cancer drugs, because they have to save the life of the patient first. Therefore, until recently, these studies were under so-called taboo. However, recently, the situation began to change. Today, there is a group of selected oncologic diseases which are considered to be treatable in principle. Those patients who several years ago were considered hopeless, today not only keep living, but also return to an active and a great life. In this context, in the clinics, they have started actively to carry out monitoring of the effects of reproductive toxicity of chemotherapy. Large enough selection of pregnant women after chemotherapy and their children were examined. At present, the progress in oncology sets a new task to realize the reproductive function in oncological patients after complete remission. Today, the duty of every physician is to inform the patient about the potential reproductive toxicity of cytostatic chemotherapy and suggest the methods to keep reproductive function active. In this report, we focused on the impact of reproductive toxicity of cytostatic drugs in women. Why women? The thing is that the large number of cancer pathologies, which are now successfully treated, are diseases of female reproductive organs. Moreover, success in treating is achieved not only due to the new effective anti-cancer agents, but also due to progress in conserving surgery of such pathologies as cancer of ovaries, Complete remission is achieved also due to the new methods for early diagnosis, for example, breast cancer. So, about what the doctor must inform the woman before treatment. How toxic is the used type of chemotherapy for gonads? How are the desirable effects? How can cytostatic effects affect the status of unborn child? And how to protect women's body against the effects of reproductive toxicity? It is known that chemotherapy causes infertility in women only in 30 to 70% of cases. Clinically, it is difficult to assess the toxicity of particular drug because the patient is injected, as a rule, with several cytostatics. We have carried out a comparative evaluation of reproductive toxicity of different types of drugs. As you can see, some of them belong to different classes of chemical compounds, but some drugs are similar in chemical structure. These drugs were selected because they are among the most popular drugs in oncological treatment. Assessment of reproductive toxicity was performed late after the introduction, and the experiments were performed with Wistar rats. To assess the toxicity to ovaries, the ovarian reserve must be estimated. Usually it is estimated by several indirect indicators. Clinically, experimental studies allow us to do this directly by counting the number of follicles at different stages of maturation. For this, we did histological sections of ovaries through the entire body, and then we have scanned and counted the number of structural elements. The calculation showed that the ovarian reserve was reduced under all kinds of influences, which is quite natural. However, the degree of damage varies significantly. The most expressed decrease in number of facilities were detected under introduction of pharmarubicin. Therefore, if the treatment includes this drug, it is obvious that in this case the most early menopause is expected. Then we have evaluated reproductive status of the female rats of the experiment. They have mated with intact male rats. I have to note that in the long-term period, 6 months after the beginning of the experiment, the most females kept their fertility. In this context, it's clear that when the pregnancy is unwanted, the contraceptives should be used. Okay. While maintaining the ability to conceive, the ability to keep the pregnancy was decreased. It was indicated by embryonic in rats growth on the background of all drugs. This data agrees with clinical observations showing that the number of spontaneous abortion is increased in women after chemotherapy. The highest embryonic death was found after use of etoposide and paclitaxel. Taking into account the increased embryonic mortality, mortality, it should be noted that there are some difficulties in extrapolation this data to human. Taking into account that the rodents have multi-fetus pregnancy, while women have a single one, the clinical figures will not be so high. Then we have evaluated the index of survival of the posterity and state of internal organs of the fetuses. It was found that the low survival rate and increased number of fetuses with changes was observed of the offspring female treated with etoposide. The toxic effect was more expressed on the background of use of etoposide. Certainly, the toxic effects of drugs may be associated with the induction of cytogenic damage in all sites. However, they also may be caused by long-term effects of drugs on normal tissue. The results of clinical and experimental studies suggests that, firstly, late after chemotherapy, the insufficient functioning of chemopoietic tissue is possible. And lower viability may be the maternal anemia. In this regard, it should be noted that the inhibition of hemopoiesis is a result of cytostatic effects of any kind. And the additional loading, which is pregnancy, toxicity to blood system may be more expressed. We have investigated hemopoietic system in pregnant rats treated with etoposide. This drug was selected because, on the one hand, it leads, as shown above, to a significant increase in fetal mortality and the decreased viability of offspring. And on the other hand, it is known that etoposide has strong toxic effects on the blood system. The parameters of peripheral blood and bone marrow in the intact animal and pregnant rats were experimentally studied in dynamics through 1, 3, and 6 months after administration of etoposide. Such parameters as blood, as of peripheral blood were changed as concentration of hemoglobin, average content of hemochloride, mean corpuscular volume, amount of red blood cells. In the bone marrow, the right number of erythronormoblasts was also changed. Generally speaking, the analysis showed that introduction of etoposide and the pregnancy, which is an additional loading to hemopoietic tissue, induced development of regenerative anemia. The nature of the changes show that anemia was of hemolytic type. This, of course, could negatively affect the state of posterity. Talking about the reproductive toxicity of etoposide, we should pay attention to its long-time toxic effect connected with mammary gland. Visual inspection of female rats in 6 months after the administration showed tumor formation in half of the rats. Histological analysis showed that breast cancer diagnosed in 50% of rats and benign tumor in 7%. I have to say that the induction of secondary tumors is one of the most serious long-term effects of cytostatic drugs. Analysis of the survival rate of posterity of female rats receiving the drugs show that it's often detected disorders of the immune system. State of protective functions of posterity depends on that of the mother. Therefore, we have investigated the cellular and humoral immunity in rat groups, which were born at the age of 25 days. To reduce the toxic effects, should to avoid the most toxic drugs as far as possible. At present, hypothalamic regulators of pituitary function are used widely. One of the problems is the need to apply long before chemotherapy. Experimentally, it is shown to be effective. Sphingosine monophosphate, which limits apoptosis in oocytes. We have studied the possibility to reduce the toxic effects of posterity by controlling the function of intracellular regulatory system. To do this, we used the official drug, glataxime. It is prescribed for treatment of the cancer patient. Glataxime restores the sensitivity of cell receptors to regulatory and transport molecules of peptide nature. This defines its immunomodulating, chemostimulating, and toxic modifying effects. The results showed that the use of glataxime reduces the number of external and internal abnormalities of the spleen. Thus, our research suggests that it's necessary to, wherever it's possible, to avoid treatment with inhibitors of topoisomerase activity and taxanes. If pregnancy occurs, spontaneous abortion is possible. Development of hemolytic anemia in pregnant women is also possible. And treatment with inhibitors of topoisomerase activity may cause secondary tumors. The born children may have decreased viability and inhibited cellular and humoral immunity. Application of glataxime can reduce the appearance of defective posterity. Thank you.
Okay, we have a few minutes for questions, if anybody has any questions.
Because I'm not the author, I know English, but the author knows the subject. I propose to ask questions in the break.
All right, that'll
I will
work.
insist.
Okay, thank you. Okay, our next presentation is it? Tell me with your last name. Pereira? Okay.
Similar. Pereira. Yes.
Right. Okay. We'll let her get started. Thanks. Is there?
Yeah.
We have this is the right one, isn't it?
Where can I find try the tox thing one? Someone can just recommend on the S.
Yeah. Yeah. That's it. Yeah. That. Yeah.
Third one.
Yeah. The mercury. Yeah.
Study aims to contribute to fill this knowledge gap regarding the toxicity of mercury in high. We work with a wild fish that is Lis aurata, and we aim to, we combine the assessment of biochemicals endpoints and accumulation of mercury The biochemicals endpoints were related with oxidative stress and also with neurotransmission. So fish have an important role in signaling water pollution once they react with a great sensitivity to changes in the aquatic environments. They are also key components of trophic change. In this work, in this study, I worked with Lisaurata, since this species is very familiar to me. I worked with this species for several times, and it's an organism that we can find in contaminated and in reference sites, in polluted and non-polluted environments. So the aim of this study was to clarify the occurrence of oxidative stress and acetylcholinesterase activity in the inhibition in the eyes of the golden gray mullet after environmental exposure to mercury. This was a field study carried out in a Portuguese lagoon, that is Aveiro Lagoon. Do you have a pointer here? Oh, sorry. This is the approach of the study. We first characterize the environmental levels of mercury, the levels that we can find in water and also in the sediment. After that, we characterize the accumulation levels in the eyes of the fish and also the ocular toxicity by the assessment of enzymatic antioxidants, non-enzymatic antioxidants, and acetylcholinesterase activity. As I previously told you, we worked in this Portuguese lagoon located in the northwest of Portugal. This is a confined area that is known by Laranja Basin. This area received for almost 4 decades emissions of mercury from a chloroalkali industry. We still can find the sediments and also in biota. So the biota still exhibit some toxicity effects related with mercury. And so we consider this area of the lagoon like a field laboratory that offers unique opportunities to study the mercury toxicity. So we went there in winter and summer 2013 to collect surface sediments, mid-water, and also to capture juveniles of the golden gray mullet. We capture these kind of samples in both sides of the lagoon, in the most contaminated area and also in a reference area. There is closest to the lagoon entrance, and we know that is not polluted by mercury, so it was selected as our reference. What we measured in the eyes of the fish were levels of mercury, both inorganic and methyl mercury forms of mercury. We also determine acetylcholinesterase activity, and we characterize the oxidative stress profile by the determination of several antioxidant enzymes and also total glutathione, and we assessed oxidative damage by the evaluation of lipid peroxidation. Next, I will show you some of the main findings of this work. I will start by showing you the levels that we found in the water and also in surface sediments, just to highlight that as we were expecting, the Laranja Basin is more contaminated than the reference site, and this happened both in winter and in summer, and we found high levels of mercury, total mercury, methyl mercury, and also inorganic mercury in Laranja Basin in both seasons. The same pattern was recorded for surface sediment, so this was this data, in fact, it confirms what we were expecting regarding the contamination of the environment. Next, here we have the levels of mercury in high wall of fish. This is box plots. I have here total mercury in the high, the white box is the reference site, and here I have the more contaminated site. I have data to winter and also to summer. Here is the total mercury, and here methyl mercury. As you can see, we found higher levels of mercury accumulated in the eyes in the most contaminated site. These occurred for total mercury, also for methyl mercury, and also for inorganic mercury that's not here, but I can tell you. We also found that in winter, levels of accumulated mercury were higher than in summer, and this happened because in the mercury available were also higher. And what we were, the next slide is related with some antioxidant enzymes that characterize the oxidative stress profile. Here I have catalase and superoxide dismutase. I follow the same box plots for both sites, for winter and for summer. What we found was a depletion of both antioxidant enzymes in the eyes of the fish from the most contaminated site. We didn't find any changes in the summer. Here we have some of the other antioxidant enzymes: glutathione peroxidase, glutathione reductase, glutathione S transferase, and total glutathione. In summer, we found an enhancement of glutathione peroxidase in the most contaminated site, also of glutathione reductase. No changes occurred for glutathione transferase, and total glutathione was also enhanced in the most contaminated site in summer. So what happened in terms of oxidative damage? We measured lipid peroxidation, which is related with the membrane damage of in this case of the eye wall cells. And in winter, we found an enhancement of oxidative damage. Finally, for acetylcholinesterase activity, we found an inhibition of this enzyme in the most contaminated here. And here I have again my triad approach. I like to call them like that. And I have a summary of our data. And as you can see, we found an enhancement of environmental levels of mercury, both in water and in sediments, in this site, in the most contaminated site in winter. These the bigger arrows aim to represent the differences between the seasons. Big errors means that in winter the environmental availability of mercury was higher than in summer. And this was also followed by a higher accumulation of mercury, both mercury forms in the eyes. And regarding with the biochemical endpoints, we found contrasting patterns between the two seasons. Namely, in winter, we found a depletion of antioxidant enzymes characterized by an inhibition of catalase and superoxide dismutase, and this was accompanied by a higher oxidative damage, a higher lipid peroxidation. On the other hand, in summer, we had an enhancement of antioxidant defenses characterized by a higher activity of glutathione peroxidase, glutathione reductase, and total glutathione, and also an inhibition of acetylcholinesterase. So this is, in some way, a repetition of the previous slide. It's just to highlight the big difference between the summer and the winter and summer pattern were due to a higher accumulation of methyl mercury in the eye. The specific form that was highly accumulated in the eye was methyl mercury and not inorganic mercury. So as final remarks, I would like to highlight that oxidative stress responses in Leigh aurata eye wall were able to detect intersite differences, reinforcing this area as a critical area in this Portuguese lagoon. We found a higher accumulation of both inorganic and methyl mercury forms, and these that were on the basis of a pro-oxidant challenge in this area of the lagoon. We found also winter-summer changes in the eyes of the fish that were mainly characterized by a higher bioaccumulation of methyl mercury in winter than summer, together with oxidative stress evidences. Mercury acted as an anticholinergic agent in fish eye, suggesting the disruption of these neurotransmitter isomes that could be on the basis of ocular toxicity. And as a recommendation for future studies, and this is what I would like to do in the future, is to search for structural and functional alterations in the eyes of the fish as a consequence of the exposure to mercury. At the end, I would like to thank you for your attention, and also to my Portuguese team and for my institutional support that brought me here today. Thank you very much.
We have some time for some questions. Are there any questions? Yes.
Yes, congratulations for your presentation. But
Thank
first
you.
of all, in one of the first slides, as far as I remember, you have written that these models could be considered, as far as I remember, as a good biosentinel.
Mm-hmm.
Are you totally sure about?
Yes.
Because to be considered one species as a sentinel in one ecotoxicology study, you are totally sure that the species is not migrate. I mean, it cannot migrate or migrate. And as far as I know, mullets are migratory species. And this could also explain the difference of the result between summer and winter.
Yes.
Because you have in the available, I have visited, and you have the influence of the sea.
Yes, it is a coastal lagoon. Yes, that's true.
So it is an atmosphere.
Yes, it's a coastal lagoon, you are right. So, and the fish could move in the lagoon, yes, but yes, we can have that discussion about about the meaning of this sentinel. But as far as I can know, and according to my experience also with this species in this system, we found several times it is a very good
it would be a very good
indicator of mercury,
it's very far to be
a good biosentinel. Is that what you were? Okay. Okay. Thank you.
All right, I guess we'll move on then.
Was there any mercury toxicity in that area?
Yes, this lagoon still very contaminated by mercury. Yes, because there are no direct emissions right now, but we do have high levels. We can find high levels of mercury stored in sediment, and the degradation of mercury, the time of the lifetime of mercury is very high, as you know. So
it is found only in the fish?
Sorry?
It is found only in the fish eye?
No, it was found high levels in the water and in the fish.
Do they eat that fish?
If we eat the fish, the people eat the fish. There are some people that could eat this fish, yes. Yes. It's not a commercial fish, but there are local people that eat the fish, yes. But there are biological studies in this area. I think it's something that I would like to do in the future.
Maybe they don't eat the eye.
Question over here too.
What about the other chemicals?
Yes. Generally, we use Generally, we call this lagoon as a field laboratory because the main contaminant in the lagoon is mercury. We already made a screening of the other contaminants, and the most contaminant is mercury.
Often find the interaction with PCBs and the effects of the interaction.
But the levels of PCBs in the sediments or even in the water are very low. We did that already. Sorry?
On the eye of the fish?
Not, we would like to move for that field of, it's something that I would like to do in the future, yes. Because there are few information about the effects. That it's not an organ that ecotoxicologists look for, so there are no works in here or in other systems. But thank you.
All right,
Thank you very much.
All right, our next presentation will be by Dr. Aburawi. Thank you.
Okay, my research about the male infertility, and we tried to use selenium to find out if it has a prophylactic or curative effect toward formaldehyde-induced toxicity. So male infertility, it means that inability of a couple to have children during or having pregnancy during the first year. And this problem is a psychological and social stress on the parent. We induce the toxicity by formaldehyde. Formaldehyde is a simple aldehyde, and it's not used medically anymore, but it is used now for cosmetics, for ladies, and in laboratory, and it is considered as a precursor for some chemicals. Previous work showed that it produced pathological changes in seminiferous tubules in male animals. We used selenium to find out if it has a protective, as prophylactic or treated treatment for the damage that produced by formaldehyde. Selenium is an essential mineral, and it's present in some foods, and it is important for a number of biological processes in the body, for human and animal. Selenium as antioxidant, it has the property to scavenge the free radicals that produced in the body during the biological process. and it is important for the important for the body to protect the DNA and to prevent the oxidative stress to produce any damage to any type of tissue in the body. Also, selenium is important for the sperm function, and it was observed that in selenium deficiency model, they observe problems in the reproductive system in mice and rats and other type of models. For that, our aim of the work is to investigate if selenium has any protective or prophylactic effect toward the infertility that induced by formaldehyde using albino mice. So we used 5 groups. The first group, we inject intraperitoneal water for injection for 5 days, and then we had our score. The second group, we inject selenium alone as a control, and the third group, we inject formaldehyde also for 5 days, and then we had the score. The first 3 groups is considered as a standard or control for the group number 4 and 5. Group number 4, we inject formaldehyde, and in the same time, we inject selenium to find out if selenium has a prophylactic effect during the damage that occur, also for 5 days. And group 5, we inject formaldehyde to produce the damage, and then we start for another 5 days selenium to find out if selenium can treat the damage after it occur or not. The score, we scored the sperm count, and we used hematocytometer slide and the morphology of the sperm, and we have to make a film on the slide and dry it and see under the microscope. Oh my God. About the discussion, here we can find that the sperm count, the group that treated with selenium alone, the number of sperm counts increased compared to the control, selenium by itself. But the others, and here we can find that formaldehyde did not change the sperm count. It produced damage, but doesn't change the sperm count. And the rest of the groups nearly like the healthy group. Here, looking for the motility, we can find that the selenium group, treated group has higher motile sperm compared to the healthy one, group number 1, and compared to the other treated group. Also here, the formaldehyde treated group did not change the motility of the sperm. Here, looking for the healthy, about the morphology, the healthy and unhealthy. We have to compare it to the formaldehyde group, treated group. Here we can see that, the unhealthy number of sperm is higher in formaldehyde compared to the rest of the treated group, while the healthy is lower compared to the rest of the treated group. So here, to find out the percentage, the unhealthy is lower compared to the rest of the treated group. Here, the motility, we find that the has a higher number of sperm that did not change the motility. Score the motility and reproductive system into the histology department. Here we can find the first of spermato seminiferous tubules, and the spermatogenic cells nearly normal, and but still there is a little bit detachment from one place to another, but we can say that it's nearly back to normal. Here also in this slide the Sertoli cells was shown, and this cells is considered normal, and it is responsible for the nutrition and support. It acts as a supportive function for the seminiferous tubules. Group number 5 that treated with formaldehyde to induce the damage, and then we treated for another 5 days with selenium after the damage occurred. Here we can see that the nearly normal seminiferous tubules, but with the higher magnification, we notice that still there is some detachments of the spermatogenic cells, but in general, it is nearly normal. So we use formaldehyde to produce toxicity in male because it's easy observable and less invasive compared to the female. Formaldehyde produce free radical, and this will produce oxidative stress on the tissue, mainly in the reproductive tissue. And also it decreases the level of the antioxidant enzyme like super dismutase and glutathione peroxidase. Formaldehyde is considered as genotoxic drug because it produces chromosomal aberration and DNA breakage and DNA protein cross-link, and it produces methylation to DNA, and this leads to gametogenic defect. These two enzymes is important for the maturation of spermatogenic cells and tests and spermatozoa, and also the energy needed for the metabolism. Formaldehyde decreases the succinate dehydrogenase enzyme, and it was found that there is a correlation in the activity of succinate dehydrogenase enzyme, positive correlation with the sperm cell counts and negative with the abnormal rate of sperm head. Formaldehyde decrease the testosterone level, and as we saw in the slide that the Lech cells was completely destroyed. That's why in the previous work the level of testosterone was less than the normal. There is another thing that formaldehyde may produce the damage through the activation of necrosis factor KB, and this necrosis factor is important in releasing it is considered as pro-inflammatory, and it releases inflammatory mediators in the body. For selenium, selenium is present in every organ, mainly the reproductive system, and it is present in high concentration in the testes. And it was observed that in selenium deficiency rat, there is flagellar disorganization during spermatogenesis, and post-testicular sperm developments were observed. Also, it was observed in selenium deficiency model animal, there is sperm defects during the spermatogenesis and maturation in the epididymis. Selenium is considered as antioxidant, so that's why it may relieve the oxidative stress that produced by formaldehyde. And formaldehyde produce free radicals in the tissue, and that's why it produces the damage. Also, selenium amyloidated DNA protein cross-link and attenuate the activation of necrosis factor KB. So we can conclude that formaldehyde toxicity was observed in this work It's a very good model for degeneration and damage to male mice genital system. Selenium by itself increase the sperm count, increase the by itself, but in combination with formaldehyde, it has a prophylactic and curative effect against formaldehyde-induced genital system toxicity. For the future work, we're going to try to find out if selenium produce this prophylactic and treatments just because of antioxidant mechanism or it has another mechanism. So we are going to repeat the same work for the female and to find out if the damage that produced by formaldehyde in DNA is transferred to the second generation. And thank you.
All right, we have some time for a few questions. Are there any questions?
Good presentation.
Thank you.
Incidentally, formaldehyde, when it is injected to rats, it produces blindness of the rats.
Formaldehyde.
Notice?
We didn't check this.
We didn't check.
We have to note this next time.
Okay, sorry. I had an observation. In one of your slides, it was shown that the sperm count was increased by selenium on its own.
Yeah,
and yeah.
The histology?
No, in sperm count. Sperm count.
Sperm count, okay.
Back.
Okay.
Okay, no.
No, metallurgy.
Back. One more. The first slide, yeah.
Mm-hmm.
So I can see the selenium caused a significant increase in sperm count, and formaldehyde on its own did not affect the sperm count.
Formaldehyde, no,
did not.
Yeah.
Now, the combination of selenium and formaldehyde ended up.
The first one, prophylactic. Yeah, the first one, number 4.
Yes.
Yeah.
It ended up now reducing the sperm count.
No, it is like the normal, but there is significant difference between this group, which take the formaldehyde and selenium together, and selenium, right? Maybe because of per each group we had 8 animal.
I'm comparing it to the formaldehyde only group.
No, there is no difference. Statistically, there is no difference.
Oh, okay.
Yes, we have a question farther in the back.
Firstly, thank you for good information. I believe this work to need in future study of the glutathione S transferase, this detoxification enzymes. Observation the role of the selenium effect of the detoxification. Okay.
Yeah, it is a part. Selenium is a part of this enzyme. This glutathione reductase, it's part of it, selenium, and this enzyme is considered as endogenous antioxidant. Yeah, it's another idea to measure it, yeah, its activity.
Thank you.
Thank you.
All right. All right, we have one more presentation this afternoon. Dr. Emi Julu, if I pronounced that correctly.
Okay, good afternoon, everybody.
My presentation this afternoon is on the renal and hepatoprotective effect of Ivingia gabonensis juice on sodium fluoride-induced toxicity in rats, Worcester rats. Where do I touch? Okay, thank you. Okay, so we try to look at the effect of this juice. A lot has been done on the Ivingia gabonensis juice, on the Ivingia gabonensis, but mostly on the seeds and the stem back of this plant. And it has been widely researched. And the plant itself consists of both the fruit, which is very succulent, and has malleable kernels from which you make the popular draw soups that is very popular in Nigeria, in Africa, but particularly in Nigeria as ogbono soup. And then it is also called dika in Cameroon. So a lot has been done on the seed. That's the seed and the kernel. And it has been shown that it has a number of nutritional benefits, and it has a lot of medicinal value. It is anti-diabetic, it is also antihypertensive, and most importantly, helps to reduce obesity. It has weight reduction tendency properties. That is the dika nut, or the not the kernel itself or the seed. Then Ivingia gabonensis is commonly known as wild mango in Africa, but in Nigeria we call it ugiri in Igbo, and then dika in Cameroon. It is especially valued because of the fat reductive tendency that it has, and it is used. A lot of studies have shown that it helps to reduce obesity. Now this is what the fruit looks like, the Ivingia gabonensis fruit. But not much has been done on the fruit itself, and this plant is easily grown, but a lot of people do not eat it. They do not patronize it. They call it people neglect it in Africa. But they use the seed, commercialize the seed and market, but the fruit is often neglected. Although sometimes it has a sweet, bitter taste. Probably that was why it is not commonly eaten. People prefer ordinary mango. So now we try to look, a lot of studies have been done on the seed and nut, like I said earlier, the references are there. And then sodium fluoride also, because we try to look at the sodium fluoride-induced toxicity. Because, of course, sodium fluoride is a well-known toxicant with all these references. But we have tried previously to look at the hypolipidemic effect of this Ivingia gabonensis juice. And we discovered that it has hypolipidemic effect. And so we now went forward to look at whether it has any protective effect on the liver and the kidney. We use sodium fluoride because a lot of debate is going on now as for the fluorosis, whether sodium fluoride could still be fluoridation, should still be used in fluoridation of water or not, and a lot of consumption by, in fact, there is a study that discovered a lot of African children are affected by sodium fluoride toxicity and fluorosis, probably because of the genetic constitution. And I know a lot of children consume pastes that is made with fluorides. So we try to look at this, the effects of this particular Ivingia gabonensis on the sodium fluoride toxicity. The experimental design, we used a total of 24 albino rats, and we divided them into 4 groups of 6 animals each. Now, the first group, which we refer to as the normal control, received just standardized pellets and water ad libitum. We didn't give anything. any toxicant. Then in the second group, which we used at the sodium fluoride control, we gave the toxicant 20 milligram per kilogram body weight, according to the previous study on sodium fluoride toxicity, using that this particular dosage. And then the third group, we gave the sodium fluoride toxicant by gavage. It was given by intubation. And in this third group, we now included Ivingia gabonensis, which was extracted. We got the fresh juice. Each day, we will administer the juice. We get it fresh. 250 milligram will be weighed and then dissolved in 250 ml of water, and we administer to the rats. So they were taking normal pellets with water too. That's after the juice, and then we'll supplement with water. But then we also gave them the sodium fluoride toxicant by gavage. And then the last group, which we used as a standard, we decided to use quercetin plus vitamin C, which are known antioxidants. The 50 milligram per kilogram body weight of quercetin and 100 milligram per kilogram body weight of alpha tocopherol. And we administered these things for a period of 35 days, after which the animals were left for 24 hours, and then we anesthetized them with dichlorometine and killed by cervical dislocation and collected blood samples by cardiac puncture. Then we looked at the liver enzymes, bilirubin, total protein, and the liver parameters, and as well as the kidney parameters and electrolytes using normal procedure. Then we also screened the fruit itself. We carried out phytochemical screening on the fruit, and it was discovered it has a lot of phytochemicals, alkaloids, flavonoids, phytosterols, phenols, phlobetanins, tannins, and saponins. This all corresponded with previous works on the other parts on this north and stem back of Ivingia from previous studies. Now, the results we obtained under the alanine aminotransferase, which is a liver enzyme, you can see that the toxicants led to highly elevated level of alanine aminotransferase, which was reduced by both the juice and the standard. And although, the reduction, it was quite significant in both the juice and the standard. Now, for aspartate aminotransferase, there is also an increase in the enzyme, which was reduced by these two. Now, the total bilirubin was also elevated in the toxicant group, which was reduced by the juice as well as the standard drug. And here, it was observed that there was no effect on the total protein. There was no increase or reduction, rather, in the total protein content introduced by the sodium fluoride. And there was also no observable effect by the juice and the standard drugs. Now, here, sodium fluoride reduced the albumin content, which was elevated in these two. Total cholesterol, there was rather an increase in the total cholesterol by the toxicant, but the two of them also increase. Now, we did not go into the cholesterol phenotypes for this particular study, but previous works on Ivinga gabonensis has shown that there is increase in, there is all elevation of high density lipoprotein cholesterol. So it may be the high density lipoprotein cholesterol present in this. Of course, we know this is an antioxidant that increased the cholesterol level in that case. Then the urea level was elevated. Also, this induced elevation of urea concentration, and it was reduced, although the reduction was more observable here than in the juice itself. Creatinine level was more significant in, there was a more significant reduction in the creatinine level by the Ivinga gabonensis juice compared to the urea. Most of the electrolytes, there was no, not much effect. Potassium level was increased here, probably because, possibly because of the potassium content of the plant. Sodium concentration was elevated also in the sodium fluoride. Here, it was also high in the quercetin, but in the Ivinga gabonensis juice, there was no significant difference between the two, that of this and the elevation, in fact, is believed probably because of the sodium fluoride that was introduced here, because all of them received sodium, the toxicant. Now, chloride was massively elevated here. It is believed, we believe that it could be as a result of rapid elimination of fluoride in place of chloride here, in the toxicant that led to this massive increase, which was also observed in the other two that received the toxicant too. There was really no observable difference between these in terms of calcium concentration. And in magnesium, there is a massive elevation of magnesium concentration in both the Ivinga juice and the standard. Well, we believe that now looking at the electrolyte level, it is as if the sodium fluoride did not have much effect or influence on the electrolyte level, but the increase that is observed here in the magnesium level may be also, in fact, it is believed that the plant is a good source of magnesium. Some studies, previous studies have also observed, the study on micronutrient concentration of Ivinga gabonensis observed that it's a good source of calcium and magnesium. So from this study, we were able to conclude that sodium fluoride toxicity caused significant increase in markers of renal and hepatocellular damage, particularly the liver enzymes and bilirubin, which were reduced by treatment with both Ivinga gabonensis and the reference standard, indicating potential protective effect of the fruit juice. The observed protective medicinal potential of the fruit juice may be attributed to the presence of the beneficial secondary plant metabolites, that is the phlebotannins and the phenolics and flavonoids that were present in the plant. So we believe that this Ivinga juice is equally good, and people will be encouraged to go on taking the juice in as much as the nuts and the stem backs are good sources of nutrients and have good medicinal value. The fruit can also be consumed too. Thank you.
All right, we have a couple of minutes for questions. Are there any questions? Yes, we have one right here.
Has this fruit been used in your country a long time?
Yes, but not a lot of, it's not very common. It's one of the fruits that are not easily domesticated. People neglect it a lot, but it's there and people consume it
And this is the
occasionally.
first scientific study like this? Is yours the first scientific study like this?
No, there have been studies on the nuts of this particular plant and the stem back, but not on the juice.
See.
Yes.
All right, thank you very much.
Question over here.
Thank you. The question is about the standardization of the juice itself. When you use it for your experiments, have you standardized the procedure or the juice itself? You can get the fruits at different times of the season.
Okay.
For example, different concentration and different treatment.
The concentration of the juice.
Juice.
I didn't understand your question, sir.
You used the juice as a drug, for example, right? But is the juice production standardized?
Okay, we just extracted it fresh. I showed the diagram of the fruit. We just got the fruit, peeled it, and then used water to extract, blend, and then administer to the rats. So nothing was mixed with it. It's the raw juice.
I think his question was probably focused on, is it as concentrated in the summer as it is in the winter when you extract it?
Okay. Summer and winter. Well, we have dry season and wet season, and this fruit stays there all year round.
So you don't see any seasonal variation as to how sweet it is or how juicy it is?
Okay. I think it is juicy all year round. Whenever you get it, it has juice in it. It doesn't dry up.
The components varies upon the time.
I don't know.
Because the components of the juice can also
Change.
along the year. I think
Okay.
Yeah.
Okay. It's okay. Okay. I don't know if it answers the question. They said it's seasonal.
All right, I think what we'll do is we'll go ahead and take the break. Right now, it looks like we're running about 20 minutes behind, so I'm going to suggest that we come back here and we'll start the next presentation at 4:15.
10 minutes also we'll do coffee. 10 minutes we'll do.
Sit there and this is when yours. I figure we could shave 5 minutes off what we were
everybody. We're ready to start.
Good afternoon, everyone. It's my pleasure to have this opportunity to introduce our work on nanotoxicology. The topic of my talk is multi-word carbon nanotube-induced lung inflammation and fibrosis. I'm going to give a brief introduction on carbon nanotubes and then talk about our studies on the pathological effects of carbon nanotubes on mouse lungs, as well as one of the underlying mechanisms we recently identified. Right now moving. I can now move
Hit the space bar.
Space, okay. So now, not doing anything. Okay. Carbon nanotubes are made of one-atom thick carbon walls that roll into long and hollow nanostructures with either a single layer or concentric multiple layers, called as single-wall CNT or multi-wall CNT, because of their outstanding physical-chemical properties, such as the tensile, electroconducting, and thermoconducting properties. CNT have been developed with a variety of applications in both industrial and consumer products.
Click with the left mouse.
The left. Okay. And the major source of CNT is industrial manufacturing. The production activity leads to a direct occupational exposure to humans, a release of CNT to natural environment, and an accumulation of CNT in food web, since CNT are resistant to degradation mechanisms. Humans can be exposed to CNT through inhalation, skin absorption, ingestion, or the use of CNT-carried drugs. The initial concern over the health impacts of CNT exposure stemmed from the notion that most CNT are respirable fibers with a high aspect ratio and are presumed to have substantial biodurability and insolubility in biological systems. And these attributes demonstrate the potentially linked CNT to toxic fibers with fibrotic and tumorigenic effects, similar to asbestos. Indeed, a variety of CNT-induced effects have been identified in recent studies performed in cultured mammalian cells and in experimental animals, including cytotoxicity, inflammation, fibrosis, genotoxicity, tumorigenesis, and immunotoxicity. And these effects demonstrate the potential health impacts of CNT on humans. And in the first part of my talk, I'm going to introduce the pathologic and molecular analysis of multi-wall CNT-induced fibrosis and inflammation in mouse lungs. Formation of fibrosis and the accumulation of collagen fibers were observed as early as day 1 post-exposure to CNT, became progressively more prominent on day 3, and reached a maximum level on day 7 after CNT treatment. And thereafter, the changes were slightly reduced, but remained significantly elevated through day 14. And we quantified the extent of lung fibrosis using a modified Ashcroft score, which indicates the severity of lung fibrosis, and confirmed this finding. And lung fibrosis is often marked with elevated expression of fibrosis marker genes, such as COL1A1, COL1A2, and FN1. COL1A1 and COL1A2 encode the chains of collagen 1, and FN1 encodes fibronectin. And the mRNA expression was significantly induced by multi-wall CNT on day 3 and day 7 post-exposure, detected by real-time RT-PCR. The protein levels of collagen 1 and fibronectin were dramatically increased in multi-vicinity exposed lungs, especially in the regions of fibrotic foci detected by immunohistochemistry. These results demonstrated elevated expression of fibrosis marker genes was directly involved in the progression of lung fibrosis induced by multi-vicinity. And in addition to the remarkable fibrosis observed in multi-vicinity exposed lungs, prominent acute inflammation was observed and characterized. The BAL fluid from inflamed lungs contains inflammatory cells and proteins that reflects the inflammatory and injury status of the lungs. Time course analysis of BAL fluid indicated multi-vicinity significantly increased the numbers of total cells, macrophages, neutrophils, and lymphocytes, as well as the level of lung injury indicator LDH activity. And next, we studied inflammatory cells in lung tissues by immunohistochemistry. Macrophage infiltration was observed at all the time points, while neutrophil infiltration was significantly induced on day 1 and day 3 and post-exposure to CNT. Furthermore, the expression and the secretion of pro-inflammatory cytokines, such as TNF-alpha, IL-6, and IL-1 beta, were dramatically increased in multi-vicinity exposed lungs, determined by real-time RT-PCR and ELISA. Taken together, these data indicated multi-vicinity potently induced acute inflammatory response in mouse lungs. And the pro-fibrotic cytokine TGF-beta1 and growth factor PDGFA have been demonstrated to play critical roles in promoting lung fibrosis in both humans and in other experimental systems. And here it's very important that we found the protein levels of TGF-beta1 and PDGFA were dramatically increased in multi-vicinity treated lungs, detected by immunohistochemistry and ELISA, and further confirming that multi-vicinity induced fibrosis has the potential to serve as a new animal model and to study lung fibrosis. And in summary, I hope I have provided some evidence supporting that multi-vicinity induced rapid onset lung fibrosis and trigger acute pulmonary inflammation and increase the production of pro-inflammatory and pro-fibrotic cytokines and growth factors. And therefore, multi-vicinity represents a potent lung fibrogenic agent, and multi-vicinity exposed mice can be used as a new animal model to study lung fibrosis in general. And in the second part of my talk, I'm going to introduce one of the molecular mechanisms underlying multi-vicinity induced inflammation and fibrosis, which involves the transcription factor NRF2. Under normal conditions, NRF2 is constantly ubiquitinated through KIP1 in the cytoplasm and degraded in the proteasome. Upon exposure to electrophiles or oxidative stress, KIP1 is inactivated and NRF2 is stabilized. Consequently, NRF2 translocates into the nucleus and activates the transcription of a variety of cytoprotective genes, through which NRF2 exerts a protective function against xenobiotic and oxidative stress. Staining of collagen and fibronectin demonstrated a detectable fibrotic lesion in control NRF2 knockout lungs, but not in control wild-type lungs. Multi-vicinity exposure resulted in a higher level of fibrosis in NRF2 knockout lungs than in wild-type lungs, especially in the regions outside of fibrotic foci. I'm sorry. Okay, sorry about this. And the lungs of NRF2 knockout mice exhibited an apparently increased cellularity, suggesting there was elevated inflammation in control NRF2 knockout lungs, but not in control wild-type lungs. Upon exposure to multi-Watson T, formation of inflammatory foci occurred in both wild-type and NRF2 knockout lungs, but the lesion was dramatically more significant in NRF2 knockout lungs than in wild-type lungs. And the numbers of granulocytes and macrophages were significantly higher in NRF2 knockout lungs than in wild-type lungs under a basal condition. Multi-Watson T significantly increased the numbers of granulocytes and macrophages in both wild-type and NRF2 knockout lungs, but the increases were more dramatic in NRF2 knockout lungs than in wild-type lungs. And taken together, this data demonstrated an NRF2-dependent suppression of inflammation in mouse lungs under both physiological and multi-Watson T challenged conditions. The phenotype of NRF2 knockout lungs indicated NRF2 might function in multi-Watson T-induced pathological effects. And we then examined whether NRF2 could be activated by multi-Watson T. Immunoblotting showed multi-Watson T significantly increased the protein levels of NRF2 in wild-type lungs, suggesting the activation of NRF2 signaling by multi-Watson T. As expected, no specific NRF2 band was detected in control or multi-Watson T treated NRF2 knockout lungs. We then examined the protein levels of two prototypical NRF2 target genes, HO1 and NKO1. These two proteins play important functions in the body's defense against oxidative stress. Here we can see the expression of HO1 and NKO1 in wild-type lungs was nearly undetectable, but was significantly induced by multi-Watson T, especially in the regions of fibrotic foci. The induction was minimal in NRF2 knockout lungs compared with wild-type lungs, suggesting the induction of HO1 and NKO1 requires the presence of a functional NRF2. And this data demonstrate NRF2 is activated by multi-Watson T in the lungs. And exposure to multi-Watson T is associated with increased production of reactive oxygen species, and oxidative stress has been implicated as one of the major mechanisms of multi-Watson T toxicity. We then examined whether NRF2 functions in multi-Watson T-induced oxidative stress. First, we determined RS production in alveolar macrophages by DHE staining. And the alveolar macrophages from control wild-type lungs did not show remarkable positive staining for DHE, but those from control NRF2 knockout lungs exhibited a higher number of alveolar macrophages positive for DHE fluorescence, suggesting the alveolar macrophages have elevated RS production in the absence of NRF2. multi-worsen significantly increased the numbers of RIs producing alveolar macrophages, as well as the average level of RIs in these cells in both wild type and NRF2 knockout lungs, but the increases were more dramatic in NRF2 knockout lungs than in wild type lungs. And next, we studied the levels of 3 oxidative stress markers, including DNA oxidation indicators 8-OHDG and gamma H2AX, and lipid peroxidation marker 4-HNE. All these 3 markers had dramatically increased the level in control NRF2 knockout lungs than in control wild-type lungs. Multi-worsen significantly increased the production of these oxidative stress markers in both wild-type and NRF2 knockout lungs, but the increases were more evident in NRF2 knockout lungs than in wild-type lungs. And these results consistently demonstrated there was a low but detectable level of oxidative stress in control NRF2 knockout lungs, but not in control wild-type lungs under basal condition. And multi-worsen stimulates RS production and induce a range of oxidative lesions in both wild-type and NRF2 knockout lungs, but the increases were more evident in NRF2 knockout lungs than in wild-type lungs. And taken together, our data suggested NRF2 has a basal activity and functions in maintaining the homeostasis of the lung under physiological conditions. NRF2 is activated by multi-worsen and plays a critical role in the defense against multi-worsen induced oxidative stress, inflammation, and fibrosis in the lungs. And NRF2 might function through suppressing multi-worsen induced oxidative stress. And in conclusion, our studies demonstrate multi-worsen induced rapid onset fibrosis and acute inflammation in the lungs, and therefore multi-worsen behave as a potent lung fibrogenic agent. And meanwhile, multi-worsen induced lung fibrosis shares a number of features with prototypical lung fibrogenic agents like bleomycin and silica. Therefore, multi-worsen exposed mice can be used as a new animal model for studying lung fibrosis. Multi-worsen generate oxidative stress in the lungs. NRF2 plays an important role in the protection against multi-worsen induced oxidative stress. And this is one of the molecular mechanisms underlying multi-worsen induced inflammation and fibrosis. At the end, I'd like to appreciate Dr. Chiang Ma for his very helpful scientific advice on this work, and Dr. Steven Reynolds for his general support on this project, and our collaborators. And this work was funded by NIOSH and CDC in the United States. And thank you for your attention. Any questions and suggestions are welcome. Thank you.
Now we take questions.
How relevant do you think these studies are to human exposure?
Okay, because right now I know some branch in our institute, they already start to collect the serum samples, for example, from the workers in workplace. So eventually we are going to move from mouse work to humans, but this is kind of like a basic understanding of the human studies. Meanwhile, the dose we use in these studies is kind of like relevant to the human, to the workers who have a general exposure on daily work, and then it's based on the human exposure in the workplace. So we believe at least it has some relevance to human study in the future.
I mean, mice are always nose breathers, right? We don't just breathe with our noses.
No, it's from
It's
the.
little some complications with the interpretation.
Yeah.
Also, I didn't understand the dose that you were using. It might have been too high. I mean, even though you say that for animals, or that it might have been relevant to an occupational exposure. I mean, I didn't see what was the actual dose.
Okay, actually we have most of the studies is work on the 40 microgram per mice, that's for a single dose. But we also did a time course analysis and dose-dependent analysis. And the mice has inflammation and fibrosis phenotype at 5 microgram per mouse. That is a very low dose. So it's kind of like in the nanotoxicities on CNT exposure, it's kind of like people already set up a range of the dosage for the mouse work. Our studies is based on the lower end of the dose range. So at this dosage, 40 microgram per mouse, and even for kind of like 1 year exposure, the mice still live very well, you know. So it's not lethal, or even not sub-lethal. It's kind of like has a phenotype, but it's not lethal to the mice. Hi.
I want to ask, what is the type of occupation and environment in which we can expose to multi-volt carbon nanotubes?
Oh, okay, yeah. For the major source of the CNT exposure is occupational exposure, you know. Because there are a lot of a variety of applications of CNT in the industrial and consumer products, such as battery, you know. So, but you know, because of the products will eventually be used by consumers, so the consumers will has a chance to expose to the CNT containing products. So meanwhile, you know, in addition to the occupational exposure, we can see that there might be solid waste and liquid waste that will get into the natural environment, you know, so that may accumulate in the food web, you know, in the water system, in the air, so that may have the exposure to humans if you live nearby a factory, you know, producing CNT, because CNT is respirable fibers, you know, so if it's in the air, you have the chance to breathe them.
Okay. Because of time, so
Yes.
Dr. Dong, this is the certification for your participation. Thank you very much.
Thank you.
The next speaker is Professor Stanley. So let's welcome.
I clicked it, I promise.
Starting Toxi Day 1 window, focus on stale_toxicology 2015, selected, 19 of 20. Item 19 of 20. Tooltip, stale.
Staley, the one under there. There we go. Thank you. Yeah. Okay. Haven't seen that one before. That's a new one. All right. On that note, we're going to talk about worms.
The work in my lab has been focused on looking at a new animal model species that has some really remarkable properties that I think might be useful in looking at different ways in which we can look at toxicology studies, and particularly neurodevelopmental toxicology studies. So I've been looking at these Schmittea mediterranea planaria. Planaria are used in a lot of American high school biology classes because they're these really cool little worms. And they're very small. They're like 2 to 6 millimeters in length, very narrow, thin, flat worms, but they have remarkable regenerative properties. So if you chop off a piece of it, each piece will regenerate into an entire worm. And that includes a very primitive brain-like structure, as I'll show you in just a second. But the specific type of planaria that I'm using is one that was collected from Barcelona by Sanchez Alvarado and Newmark in 1998, and then they've been cultured in the laboratory since then. The variety that we use are asexual worms. There is a sexual variety as well, but we haven't done any work with that at this point. So the SMED planaria that we use are considered to be one of the simplest organisms alive with a central nervous system. They have this bilobed brain structure here, which we call the cephalic ganglia, this purple structure here, 2 photoreceptors at the top of the head, and then these bilateral nerve cords that traverse the body with a peripheral ladder that goes through here. So we have a central nervous system that we can look at. And remarkably, these worms, even if you cut off a very small piece of the tail, will regrow the rest of the entire worm, which includes a complete nervous system, even if it had no nervous system to start out with. So we hypothesized that we can use this as a model system for looking at neurodevelopmental processes in a very easy, quick, and potentially high throughput method or manner. And these properties are largely driven by a large collection of pluripotent neoblasts. Those are their stem-like cells, and they're pluripotent, and they exist in 10 to 30% of the cell population. So they are chock full of pluripotent stem cells, giving us a really into entire worm. I have succeeded in cutting it. 1/279th can regenerate into an entire worm. The genome for the SMED planaria that we used has been sequenced and annotated pretty well. It's available through Sanchez Alvarado's lab's website, and it's maintained there. They've also been used for various behavioral teratology experiments, so looking at different drug combinations, for preliminary pharmacological experiments, looking at cocaine and marijuana substitutes and opioid dopamines, for withdrawal effects and dependence in those different things. Forward. So our hypothesis that underlines all the experiments that we do is that a head regenerating planaria, or planaria where we've taken the head away and it will now be recreating a new brain, can be used as a viable model system for investigating direct effects of toxin exposure during neurodevelopment. So that process of head regeneration is akin to a neurodevelopmental process, and if you put toxins, if you expose that neurodevelopmental process to toxins, you can start to begin to draw testable hypotheses about the neurodevelopmental processes and the effects of that toxin exposure. So I think, like Mike was saying earlier, we can't just ask these guys how they're feeling or what they can think of and how their brains are working. So we're actually doing a 2-part test in order to test the cognitive functionality of these animals. One is an assessment of their movement. So these are bottom dweller animals that live in fresh water. We actually store them in Tupperware containers in the lab. They're really amenable to my undergraduate student research laboratory. But they swim away from the light because the light means predators. So they have no negative phototaxic behavior, but in order to swim away from the light, they must be able to swim. So we first test their ability to move, their motility, using a planarian locomotive velocity test. And this was developed by a pair of pharmacologists at Temple University, where they look at actually a minute-by-minute progression of the worm's ability to cross grid lines within a 35-millimeter Petri dish. So this is a 35 Petri dish, and these are half-centimeter squares, and you just follow the planarian's head as it moves around and get a measure of their motility. We've done this in the presence of alcohol. Our preliminary studies in looking at this as a model system were in the effects of alcohol, because we know alcohol has drastic effects on the neurodevelopment of humans. So we looked at the difference in the number of lines crossed for worms that were in a control solution, worms that were in a 1% volume by volume alcohol solution, and worms that are in a 2% alcohol volume by volume solution. And the fact that this is a dose-respondent curve indicates to us that alcohol is entering our worms and having some kind of effect, which is always something that's a little bit in question when you put something just in the water and you're expecting it to diffuse into the animal. And the second property that we looked at was their ability to swim away from the light, their light avoidance behavior. So we call this our planarian light avoidance. And this was a method that was developed in 2004 for looking at the recovery of the photosensing system during the head regenerative process. And you can see that in these studies that were published in 2004, they regain the ability to swim away from the light in about day 5. And that's measured by putting the worm in the start area and then calculating the amount of time they spend in the darkest quadrant of the testing chamber. So we just give them a 90-second test where we quantify the amount of time that they spend in the darkest quadrant. And when we do this with our alcohol-exposed worms, we can also see a similar pattern where the controls spend the most amount of time in the darkest quadrant. 1% alcohol diminishes that, but not in a statistically significant way. These are 95% confidence interval bars. But the 2% ethanol, which also reduces their motility, if you remember, is going to also reduce their amount of time in the darkest quadrant. But the problem is, these are two disparate tests, and how do we really take one into account when trying to identify their overall cognitive function? So what we've done is we've developed what we've called a cognitive function score, which is a weighted average of their light avoidance behavior based off of their ability to move. So we've taken the percentage of time that they spend in the darkest quadrant and divided it by the number of lines crossed. So it's a movement normalized light avoidance behavior score. And here you can see that these scores are not any different statistically not different for the control and 1% exposed alcohol. That's a 1% volume by volume solution. But the 2% does create this pretty much ablation of movement. These worms are pretty sedated. They don't go anywhere. With the exception of a couple who do very well and then throw our error bars to be very large. But what's interesting about this is these are intact worms. These are worms that have never been cut. They're just sitting in the solution for the entire 9 days. The interesting thing comes about when we actually do what happens in regeneration. So just to keep this in mind, as we look at the regenerative properties, we saw that high doses inhibit the cognitive function in planaria, so therefore we won't be looking at the 2% anymore because it's hard to measure their cognitive behavior when it's also inhibiting their ability to move. But the lower doses do alter the cognitive function by slightly attenuating their cognitive function, but not in a statistically significant manner across the path. So what happens in regeneration? Well, we're going to cut off the worms' heads on day 0, and there's no indication that these worms feel any pain. We've not yet recorded that, so and both pieces are going to grow back to be healthy adult worms. So it sounds bad to say I'm cutting off their heads, but I'm going to cut off the heads at day 0, and then this is the typical path that a control worm will take, the average trajectory across time. So in the first 8 days after head resection, you see the cognitive function score gradually increase. And that corresponds to the reacquisition of photoreceptors in this blastema area, this light transparent area. The full development of the photoreceptors, but not yet the ability of function within those photoreceptors. It's been shown that those are there, but not actually connected to the cephalic ganglia, so it doesn't actually produce any movement associated with it. But then you have this large jump that occurs on day 6, and what we've seen where that's a fully functioning worm. When we put alcohol in, however, we see this delay. So here we're seeing the control worms go back to their, what we'll call a normal level, somewhere around day 6. It's not until day 7 when we expose them to ethanol. So although we don't know what the molecular mechanism is, there's something that's being directly affected by the ethanol in the water that's affecting that neurodevelopmental process. And so we want to extend this to both identify the molecular effects, that what's underlying this delay, as well as see if we can replicate it in other known toxins or potentially unknown toxins as well. So we said that already. So the work that we're currently doing in our lab, and this is hot off the press, we're looking at BPA as a known endocrine disruptor. So we've talked a lot about estrogenic type of molecules. BPA is an endocrine disruptor. There's a lot of controversy in the literature about its effects and how the effects manifest. There's a dichotomy in the results that we see. But we do know that BPA is transferred across the placenta. We do know that BPA has been measured in 93% of the human population. So we are getting it into our bodies somehow, and presumably that could get into the fetus. And if that would affect fetal development, that would be something that would be important to know and to be able to do. Interestingly, it's been banned in baby bottles and infant formula can linings, so although the effect hasn't been replicated, and like I said, there's a lot of controversy in that, but it is being taken away from our most vulnerable populations there. But so I'm collaborating with other engineers at Rowan to actually design new molecules that still possess the properties of BPA. So BPA is great for plastics and epoxy resins. It has properties that we want it to have. It just might not be very good for us. So can we make a molecule, can we design and engineer a molecule, excuse me, that would still maintain those properties but prevent the toxic effects? And the molecule that my collaborator has developed is this bisquioxal F, or BGF. If you go back and look at the structure here, you can see this is the structure for BPA. The chemical structure for BGF is this. Slightly different, but largely of the same character. He's making it from trees, so he's taking lignin from wood products as a waste product from the paper and pulp industry, and being able to use that to generate the BGF. So he's doing it entirely from green sources. But we don't know, we can make this molecule, it may be great for epoxies and plastics, how is it for us? And how does it affect humans? So we want to be able to try that in our worms, and the first tests that we've done are just looking at fatality. So if we put the dose very, very, very high, does it affect our worms at all? And the answer is yes. So this top graph is the fatality rate after 24 hours. So BPA, these are very high concentrations, obviously. BPA will cause this fatality in this percentage curve, but the BGF molecule, the new engineered molecule, delays that until higher concentrations. So we can use more BGF before we get the same fatality rate, which is promising. And we can also look at lethality as a time course. So here we're looking at 6 parts per million BPA and BGF. Here we're looking at 8 parts per million, and here we're looking at 10 parts per million. And you can see that in each of these cases, the BGF delayed the lethality of these compounds. So it seems to be performing better. We're in the process of doing the regenerative tests for this and replicating those so we can see how it works with the neurodevelopment. But it does give us some indication that our model system may be useful in determining toxicity of these novel compounds in a relatively straightforward, quick, and potentially high throughput manner. So if we condense that data all down to the LD50, we can look at the curves of LD50 for the time after exposure, and you can see that the BGF then has a higher LD50 at lower times, but then they do level off after periods of time. So this molecule may seem to be much better in the long run, so we may need to continue to look for new molecules for that. So these are some very preliminary results. Preliminary at this point, but they seem to indicate that we may be able to see some differences in the cognitive function there. And you can also see those occurring with the BPA and BGF when you test the different. We have 2 different levels of BGF here. The 2, which is the green line, so that's a lower line. And it does appear as though the purple and red lines, the 2 and the 5, are more similar than the 2 parts per million of the BGF, indicating that you can have a higher level of BGF before you get the same effects as you would for BPA. So that's promising in terms of determining new molecules. But what we're really excited about is that this might be a model system that we can use for preliminary testing of toxicity and neurodevelopment. So neurodevelopment is one of those processes that's very difficult to to study because there's a lot of time involved in gestational periods if you're looking at an in vivo model. Neurodevelopmental processes in vitro are not necessarily well characterized or well replicating the in vivo condition. So we've now looked at it preliminarily in the BPA and BGF context, and also somewhat more extensively with alcohol exposure. But we think that this is an animal model system that we can study direct effects of the toxins on neurodevelopment. You can also pull that down to stem cell differentiation, because remember I said that these animals are chock full of stem cells. So we can look at that at a direct effect on the stem cells themselves. And there's a high potential for these to have high throughput experimentation. So you can imagine the type of setup where you could measure 100 worms at the same time. You can have an automated system there. You could test a variety of different doses and timing exposure and combinatorial effects and those kinds of things. Within about a week, you can get the neurodevelopmental information. So we think of it kind of like a coal mine canary. So you have a canary that alerts you to carbon monoxide in your coal mine. We think that this might be able to give us some early preliminary information that a novel compound may have some neurodevelopmental effects. So with that, I'd like to thank the people who have helped me do this work, my collaborator, Dr. Stan Zione at Rowan, and Dr. Kevin Dahm at Rowan as well, and then some of my students. The alcohol work was primarily done by Jesse and Tyler. Jesse's now a PhD student at the University of Pittsburgh, and then a whole host of undergraduate students, some of whom are pictured here, who have helped do some of the testing along the way. And with that, I'd like to open it up to any questions. I'd be happy to answer any questions for you.
Neurotransmitters?
So the question was, did we try to measure neurotransmitters? And no, we have not done that. We would really like to, but I work at an undergrad, well, what has until the last year or so been an undergraduate institution. So getting the testing done and showing that the model would work was our first step. But you're right, I would love to look at the neurotransmitters. I come from a neuroscience lab, so that's definitely something I'd love to see.
Could this model potentially be used to monitor for chronic effects of exposure to organophosphorus pesticides?
So that is an excellent question. The question was, can we use the model to look at long-term effects for pesticides? The answer to that, I believe, is no, because these animals are continuously renewing themselves. The longer period of time that you wait, so if you were to look at something in a chronic condition, the more time you give them to repair. So one of the things that we found with alcohol was that we didn't see any long-term effects, even when we put them in very large concentrations of alcohol after a long period of time. The difference that we saw was in the developmental dynamics, the way that they actually redeveloped. Because we really think that at long times, their stem cell population is just turning over and turning over and turning over, and they're creating basically a new organism to adapt to whatever changes you may have. So I don't think that it would be good for chronic effects. I could be wrong, but I would be wary of that. But that's an excellent question. Thank you.
We take the next question.
The quantification of BPA and the environmental contamination is a big issue for BPA studies. I noticed the concentration you use of BPA is like 2 ppm or 6 ppm. Such concentration may be even below the environmental level. So I don't know, are you sure the concentration you use are really true BPA concentration? Maybe higher than what
Right. So you're exactly right. So some of those values that I think we went up to 20 ppm. Some of the values are quite high, but you have to remember these are the levels in the water, not necessarily the levels in the animals. So
you can even detect.
Yeah, you can. I know. We've seen it. We've seen it in the hundreds of ppms of BPA. Sometimes it's ridiculous. And presumably that's in the water that these would be swimming, the freshwater streams that they would be swimming in normally. We see them die at higher levels. And it may be our ability to suspend the BPA into the water that we're using, that it's changing the pH to a level that they're not working. But once we get to 20 ppms, all the worms are dead within 24 hours.
So we'll take the last question.
Interesting we would want to eventually relate your study to human, right? Now, with this disadvantage, replication and all that, is it possible for you to use a model that is closer to human, like the rat model? They live with us on Earth, they breathe the air we breathe, and I think there's similarities and relationship would have been closer compared to this.
No, that's an excellent point, and that's one that I always get from reviewers, and you mentioned the relatability to humans. You're entirely right. We are pretty far from humans when we're talking about here. And I think of this as very much a preliminary screen, and you would never jump directly from a worm to a human. What I see it more as is an introductory type of high throughput thing that you can't do as well with those types of model organisms, that would give you some insight into a preliminary idea of what these molecules would be doing. And I would never say that something that you got out of a planaria would really make or break something, but I don't know the false positive, false negative rates, et cetera, for activities or neurodevelopmental effects. But I think that this gives us a way to really do it in scales that we couldn't do in other laboratory models. But that's an excellent point. That's something that I struggle with all the time. So
let's thanks for the wonderful presentation.
I know I have mine. Are you up next? Oh, you're next. Okay. Hopefully it doesn't talk to you.
Mm-hmm. Where's the full screen? Huh? Oh, there.
99% available.
Push.
First of all, I would like to thank the organizer for giving us such a wonderful opportunity so we can gather together and learn from each other. My topic today is physical chemical transformation of zinc oxide nanoparticles on the aging process in aquatic environment and the toxicity to green algae. I'm from Hefei Institute of Physical Science, Chinese Academy of Sciences. As we know, nanomaterials and nanotechnology has emerged as a fast developing area of research. And the nanoparticles can undergo physical chemical transformations in the environment, and this can give rise to the unexpected environment and health risk. And in this study, I studied zinc oxide nanoparticles, and it can undergo physical chemical transformations, and so the toxicity of zinc oxide nanoparticles may be changed in the aquatic environments. But there is still a lack of quantitative assessment of the transformation of the zinc oxide nanoparticles, and there is no clear understanding between the relation between the transformation and the toxicity of zinc oxide nanoparticles under the aging process, especially in the aquatic organism. And in this study, I choose microalgae as an object because they are the primary producer in the aquatic food chain, and they can be very sensitive to contaminants. So it is a good model for evaluating the ecological impact of toxic and nanoparticles in aquatic environment. Before I introduce my work, I would like to mention that work I participate in regarding the aged, the toxicity of aged zinc oxide nanoparticles in mammalian cells. We found that in this work, we found that the cytotoxicity of a nanoparticle decrease with aging, but the genotoxicity increase with the aging. And this work has been published in the Toxicology recently. But this work was done in mammalian cell. So we launched another research regarding how this aged zinc oxide nanoparticles affected this aquatic organism. And so the outline of this talk, first we would like to show the morphological and quantitative change of zinc oxide nanoparticles on the aging process. And then we show the interaction of zinc oxide nanoparticles with the algae cells. And we give the evidence for the characterization and the quantification of the neoformations transform from these nanoparticles. And then we show the effects of the aging process on the toxicity of zinc oxide nanoparticle to green algae cells. And we discuss the mechanisms of the aged zinc oxide nanotoxicity. For the morphological and the qualitative change of zinc oxide nanoparticles, our SEM image shows that they are the spherical and the short rod shapes, but there is no obvious changes in the shape types with aging. And this TM image shows that there is sheet-like neoformations in the aging process. So what is the interaction between these nanoparticles and the algae cells with aging? And we see that these particles attach to the surface of the algae cells, and they have showed the distorted morphological features. And so what are these neo formation of these nanoparticles? So we use the XRD and FTIR Raman spectroscopy to identify the neoformations which has been transformed from the zinc oxide nanoparticles. And we see that this is aged zinc oxide, and there is zinc hydroxide and hydrosynthesides from this aged nanoparticles. And this is the Raman spectrum, and we can see there is 145. This peak, this comes from the hydrosyntheside, and this is 840. This comes from zinc hydroxide. And so we confirm that there is a transformation, and we found this substance. And how about the toxicity of this aged zinc oxide? We did experiments with 5 groups, and one is control without the nanoparticles, and another is fresh zinc oxide nanoparticles without aging. And these particles aged for 30 days, and this for 120 days, and this for 210 days. And we see that the growth rate of this algae has been suppressed significantly. And we also measured the chlorophyll A, and we found that the growth rate decreased at first until the 30 days, and afterwards this growth rate becomes increased again. And so we also measure the photon synthesis activity, and we see the similar phenomenon, and first decrease and then increase. And we measure the damage of the cells, and we see the damage increase then decrease. And also for the ROS content in the cells increase and decrease. So what's the reason for that? As we know, in the aging system, the particles, the nanoparticles can release zinc ions, and these zinc ions increase with time. And after 30 days, they become stable. The concentration of zinc ions become stable. And so this zinc, so at the early stage, this toxicity is mainly attributed to the zinc ions release. And we found that, and to confirm this, we add this TPEN, which is a zinc ion inhibitor that can, and after add this, we find that the toxicity decreased. So this confirms that zinc ions plays the dominant role at the beginning. But after 30 days, this toxicity decreased. Why? Because this is because the transformed neo formation, they are less toxic. And to confirm this, we have to measure measure the transformation of these nanoparticles quantitatively. To achieve this, we're using the spectroscopy method. And just as I mentioned, we can use Raman spectroscopy to quantitatively evaluate how much the amount of this hydroxide and the zinc hydroxide. And for this 145, this peak, we can evaluate this hydroxide. And this, for this 830, we can measure this one. And just because of time, I just want to go quickly. And if we have three mixtures with three mixtures, we do the experiment, and we can calculate how much the hydroxide and how much the zinc hydroxide inside this system. And so this is the result. As we see, within 30 days, there is no much change for this neo formation, but after 30 days, this hydroxide and the zinc hydroxide increased, and they are just less toxic. So as I just put the result together, we can see decrease, increase for the growth rate, and the photon synthetic activity, decrease, increase. And also, but the damage, increase, decrease, increase, decrease. So this constant to our assumption. At last, I want to also mention that we can use the spectroscopy to identify different types of damage of the cells. Because this FTI can identify lipids and proteins and carbohydrates, they have different bands, so we can characterize this kind of things. And this just shows the lipids, shows in this area, this region, and the protein in this region. So using the spectral method, we can investigate how much the damage inside the cells, and so we measure the using this method, we quantify how much the protein, lipid, and change in the fresh nanoparticles or aged nanoparticles. And this is identification of the damage of the cells. This is fresh nanoparticles, and this is because of the aged nanoparticles. So there, indeed, there is difference. So this is my conclusion. Zinc oxide nanoparticles undergoing aging process in water will transform into basic hydroxide, and the zinc and also release the zinc ions, and observe the toxicity both fresh and aged zinc oxide nanoparticles confirm there is significant contribution from the zinc ions. And we also see the decreased toxicity from the aged zinc oxide. This is because of the less toxic zinc hydroxide and the hydrocarbonates transfer from the zinc nanoparticles. And at last, I want to, because I'm actually, I'm very interested in how to use the spectroscopy for this different kind of researches. So this is also a demonstration how I can use the spectroscopy to study the toxicity, especially for the nanomaterials, between the nanomaterials and algae cells. So thanks for your attention.
We do have time for a couple of questions.
I would like to mention that this work, a lot of experiment work was done by Dr. Hong Zhang, a group member in my group, a member in my group.
All right, well, if nobody has any questions, let's thank him one more time for a great presentation. We'll invite our next speaker up.
Good afternoon, everybody. I'm Dr. Friday E. Ubo from University of Calabar, Nigeria. Before us today, I'm presenting a topic, comparative protective potentials of vitamins C and E against nitrocellulose thinner-induced heterogeneity in male and female albino oyster rats. There is increasing concern about voluntary and involuntary exposure to solvents that generate pollutants into the environment. And we've understood that exposure to these pollutants can occur from intentional, and of course some are unintentional ingestion or inhalation of these solvents. In the course of this, we have both acute intentional or chronic unintentional exposure to some of the industrial solvents for many that can cause many serious intoxicating and health problems. A nitrocellulose thinner is one of such solvents of abuse among delinquent children in some countries. And in the form of lagua thinner, is an industrial solvent commonly used in furniture, paints, and automobile spray painting industries. The solvents we know contains different organic substances such as ethyl benzene, toluene, and butyl acetates. Of course, these chemical substances are known to constitute chemical pollutants in the various environments. According to the report of World Health Organization 2005, some of these chemical pollutants are detectable in the household and workplace environments. Hence, exposure to the chemical pollutants from nitrocellulose thinner in indoor and outdoor environments may be common. We've understood that the possible mode of exposure to the constituents of these solvents may be from the oral ingestion of drinks and foods that are being contaminated in the course of application, and or the inhalation of contaminated air in the workplace environment, as well as dermal exposure from spillage during usage. It is a common practice in some developing countries. You see the workers that apply these solvents just using without any protective device, and in the course of this, the fallouts from these chemicals may contaminate the food and even the environmental air. And in the course of this, we see that manufacturers, furniture, automobile, and spray painters, as well as those residing around these work environments, therefore tend to be the population with greater risk of exposure. Particularly, occupational exposure to mixtures of toluene and other chemicals have been reported in painting or lacquer workplaces. And different health and environmental hazards have been reported to be associated with exposure to some of these chemical substances. Particularly, exposure to nitrocellulose thinner and other related organic solvents has been reported to induce various toxicity effects like hematotoxicity, nephrotoxicity in experimental animals and as well as human samples. The likely cause of the nitrocellulose thinner toxicity has been suggested by some of the researchers to be the interaction between the constituents of the solvent and the reactive metabolites. The major concern is that measures of protecting or ameliorating the effects of these substances is inadequate. But however, in our previous studies, we've discovered that vitamin E and C can provide protection against nephrotoxicity of the nitrocellulose thinner and the toxicity of gasoline fumes. So this study therefore assessed the comparative protective potential of vitamin C and E against nitrocellulose thinner induced heterogeneity in male and female albino oyster rats. The major reason why we decided to work on this was that we discovered that most of the people engaged in this occupation normally complain of headache, and from the epidemiological survey, most of them have hypertension, and in attempts to look into the possible mechanism of these hypertensive reports, we decided to start with looking at the atherogenic potential. We used 48 rats, 24 males and 24 females in this study. The animals were allowed free access to standard laboratory diets and tap water at the bitumen. The work was carried out under non-prevailing room temperature. From the preliminary studies on the LD50, we discovered that 30% of the LD50 exhibited the toxicity effects that were aforetime mentioned. The rats were divided into 4 groups on sex-dependent basis, as outlined. Group 1, A and B, represented male and female that served as a negative control that were given placebo, while Group 2, A and B, were given the nitrocellulose thinner. Group 3, A and B, were given vitamin C in addition to the nitrocellulose thinner, while Group 4 were administered vitamin E. The animals were sacrificed under chloroform anesthesia after 24 hours of the latest administration. And the blood samples were collected, plasma separated for biochemical analysis. To assess the heterogeneity, we used a lipid profile of the plasma. We also derived the heterogenic indices from the ratio of total cholesterol and HDL, total triglycerol and HDL, and the ratio of the difference between the total cholesterol HDL and HDL, and the logarithm of total cholesterol to HDL. the reagent kits obtained from biosystem laboratories were used in the study. Of course, all the reagents were of analytical grade. We used appropriate statistical method to validate the statistical differences between the mean of the results. The results obtained from the study showed that the levels of plasma total cholesterol, triacylglycerol, low-density lipoprotein, and the heterogenic indices recorded for male and female rats exposed to the solvent were significantly higher, while LDL levels were significantly lower compared to the negative control. And they recorded a nitrocellulose thinner induced percentage increase in total cholesterol, triacylcholesterol, the low-density lipoprotein, and the decrease in high-density lipoprotein of female rats. We have the values represented there, were however insignificantly higher compared to the percentages recorded for females. The increase in the total cholesterol, triacylglycerol, and the heterogenic indices and decrease in HDL levels reported for the rats exposed to natural thinner were therefore observed to be sex independent. Okay, this shows a picture of the results. Group 1 represents the negative control. Group 2 represents the positive control, the group that was administered with the solvent. Group 3 represents the group administered with vitamin C, while Group 4 represents the animals administered vitamin E. We discovered that from what I explained earlier, that the concentration of cholesterol was significantly higher in the positive control group compared to the negative control, and administration of vitamins E and C reduced the level of this lipid, and that was also applicable to triacylglycerol. The plasma LDL level was also increased by the solvents, and the vitamins, which of course we know are potent antioxidants, were able to reduce the level of the LDL. The HDL was significantly reduced by the administration of the solvents, while the vitamins were able to increase the level of these fragments to the level almost within the range of the control. The table shows the heterogenic indices, the ratio of total cholesterol to HDL, total triacylglycerol to HDL, and other heterogenic indices. From here, we discovered just that the results follow the same trend with other lipid fragments, indicating that natural thinner is a risk factor to atherogenesis. From the results of this study, it therefore suggests that oral exposure to natural thinner introduce some chemical agents into the body tissue, and we are on the process of looking into the specific mechanism responsible for the increase in the lipid profile and the decrease in the HDL, whether it is at the point of synthesis or at the point of degradation, and the specific role played by these antioxidants in ameliorating or protecting the seeming toxic or adverse effects of these solvents. We also see that these agents or their metabolites might have interacted with the body tissues to induce the heterogeneity in a sex-independent pattern. The results also indicated that vitamin C and E may provide protection against nitrocellulose-induced heterogeneity in male and female rats, and that vitamin E relatively is more potent than vitamin C in providing the protection effects against the nitrocellulose thinner-induced heterogeneity in both male and female rats. The results of this study, of course, correlated the results of our earlier reports of the role played by vitamin C in hepatotoxicity, effects of gasoline vapor, and the nephrotoxicity effects of the nitrocellulose thinner. And based on the results of this study, it may therefore be concluded that vitamin C and E are being potent antioxidants, are capable of providing protection against the nitrocellulose thinner-induced heterogeneity in male and female model rats, and that vitamin E showed a relatively higher potency than vitamin C in this regard. As I said earlier, we are on the process of considering the specific mechanism in order to be able to establish this heterogenic effect that is being observed, and to be able to relate that with the commonly reported hypertension by the workers involved in this work occupation. Thank you very much for your attention.
I have done some studies with vitamin E and C, and I've shown, but it's not in rats, I have shown that they can flip from an antioxidant to a pro-oxidant response. Have you witnessed that at all?
Oh, we have in other studies. That is why I mentioned that we are in the process of designing a work to look at the actual mechanism of the effects that we are observing in this present study.
So it's going very nicely in the right way. But I mean,
they are, or yes, show the effects, but the standard of your part is so high that you are not able to realize that you are just facing you. There is a big problem, there is an extremely big variation.
well, I suppose it depends on the animal and the individual or whatever. And we did it with polyphenol tea, black and green tea, and found that halfway up the dose response curve, they just flipped. And that's what can happen with some of these studies sometimes. But you haven't observed it, so that's good.
Thank you.
I have a question. Can you comment, and this is probably my naivety not being in this area, on how much vitamin E and vitamin C that is? Are you talking about massive doses, or is this a fairly normal dose for?
We apply normal therapeutic dose, or normal prophylactic doses.
Okay.
Is what
okay.
we applied.
Okay. Thank you.
Thank you.
Any question? If no question, let's send the speaker.
Do we have our next speaker? Yeah. Careful, it might start talking. I don't know what's going on with the computer. Just push the F5 button and then it won't talk to you. The button up at the top that says F5. Oh, you got it. You got it.
My name is Fort Savior from the Department of Chemistry, University of Calabar. I'll speak on serum and liver tissue hydrocarbons contents of male rats orally exposed to bonylite crude oil. We are aware that crude oil plays a very important role in world economy. And I know we're also aware of the fact that there's a drop in oil price. That does not mean we will forget about crude oil. We see that the polycyclic aromatic hydrocarbons are among the hydrocarbon constituents of crude oil that are dangerous contaminants released into the environment. And there's been a lot of studies on this. And because of oil exploration and exploitation, we see a lot of releases. Could be as a result of sabotage to the environment or due to damaged pipelines, gas flaring, and water bill. And if you look at the Nigerian environment, you look at the Niger Delta environment, you can hear about, local oil exploration, where people tamper with the oil pipelines, get the crude oils. We have local refineries, and by so doing, the oil activities becomes like a day-to-day business. And traditionally, we also see that people tend to look at crude oil as a means of offering some therapeutic effect, like normal aspirin claim it has an analgesic effect. There are some studies on that. And so the effect on this oil releasing into the environment has been so massive, there is need for us to look at the effect. There's been a lot of exposure to crude oil and the adverse effect, and there's been a lot of studies on the adverse effect of crude oil exposure. And this has shown there has been complications to several organs like the liver, the lungs, the kidney, and the skin. It has been reported that several petroleum and petroleum products induced toxicity effects are attributed to hydrocarbons constituent of these products. However, there is scarcity of information on the specific hydrocarbon constituent of Nigerian bonylite oil accumulated in the blood following oral exposures. And so the study aimed at assessing the types and concentration of hydrocarbons accumulated in the serum and liver tissues of male rats orally exposed to bonylite crude oil. We used 20 male albino oyster rats for these studies, and the rats were distributed into two groups. We had the test group and the control groups, with 10 rats in each of the groups. The test animals were given 60 milligram per kg body weight of bonylite. This was based on the LD50 that was conducted. This was administered daily for 28 days, while the control animals were given distilled water. The animals were housed in plastic cages with metallic top and were kept under prevailing normal room temperature and humidity, and on a 12-hour light-day cycle. They were allowed free access to standard laboratory animal pellets and the water. For the crude oil, normal procedures were followed. We obtained permission from the Department of Petroleum Resources, the zonal office in Port Harcourt, south of Nigeria, and we got it from the NNPC Total Shell Joint Venture crude oil line. The animals were handled and treated in accordance with the institution's animal handling ethical committee, and all the chemicals used were of analytical grade. Now, after 24 hours from the last exposure, the animals in both groups were sacrificed under chloroform anesthesia. Blood and liver tissues collected for analysis. The whole blood samples were collected by cardiac puncture, allowed to clot, and the serum separated after certification. The serum and the digested liver tissue samples were extracted and analyzed for hydrocarbon types and concentrations using the gas chromatography. The data generated from this research work were reported as the means and the standard deviations. The data were further computed and analyzed using a one-way ANOVA and unpaired student test. These data were analyzed using the statistical package SPSS, version 18.0 for Windows, and considered significant at P less than 0.05. Table 1 shows the distribution of the animals. For the results, we can see this is the control for the serum, and then we have for the liver, test for serum and the liver. This represents the benzene, and while this is the xylene. And we can see from the test we have high level of benzene and a high level of xylene. This shows the serum and the liver tissue toluene and ethylmethylene concentrations of rat oil exposed to Nigerian bonylite crude oil. This is the control, and these are the values for the test. We can see high values of toluene, both in the serum and the liver. This is for naphthalene and the B-text for controls, and for the test we can see a very high level of B-text there. And then we have the pyrene, and of course total PAH for test and controls. You can see the controls and high levels for the test. Now from the results that we've obtained, it shows clearly the distributions of the different constituents of the hydrocarbons in the different tissues, serum and the liver. The results show that benzene, toluene, ethylmethylene, xylene, B-text, pyrene, naphthalene, and total polycyclic aromatic hydrocarbons in the serum and liver tissues of rat exposed to bonylite crude oil were significantly higher compared to respectively with the concentration in the serum and liver tissues of the control rats. However, there was no significant difference between the serum and liver controls of these hydrocarbons in both the control and the test animals. And so the reports demonstrate, based on literature, that benzene, toluene, ethylbenzene, and xylene are among the major aromatic hydrocarbon constituents of most petroleum products, including gasoline. And we can see from the literature that most of these hydrocarbons are biotransformed into reactive metabolites by hepatic tissues metabolizing systems. Earlier reports also indicated that the aliphatic and aromatic hydrocarbons, which are the major constituents of petroleum products, are metabolized in the liver tissue. Several tissue toxicities have been reported for petroleum polycyclic aromatic hydrocarbons in humans and experimental animals. There's been a lot of studies on the toxicity of these various components. And so the result of this study indicates that these hydrocarbons, on absorption within the gastrointestinal tract, are relatively distributed equally within the serum and the liver tissues. The result also suggests that the hydrocarbons and all their metabolites are implicated in the different tissue toxicity effects reported for petroleum and petroleum products in humans and experimental animals. I want to appreciate the organizing committee for giving us the opportunity to make this presentation, and of course my supervisor, Associate Professor Dr. Obo, and then Professors Ebong and Eyong for their guidance and support during the course of the study.
Now we take questions.
Thank you.
Were you expecting differences between the two tissues you looked at? You said there's no difference.
We were, in terms of distribution, we were trying to see if there could be differences due to the action of metabolizing enzymes, or if there could be anything that could affect absorption, and that would result in difference in distribution. But from what we saw, there was no significant difference in distribution.
Okay. Let's thank our speaker one more time. Do we have our last speaker of the evening here tonight? Dr. Tilak? Fish and frog? Tilak. I guess we're done then. Thank you to everybody for a good day.
Thank you, everyone, for attending. If any of you drove today, please see the registration desk and give them your parking ticket, and it'll be validated, so you don't have to pay for parking. We begin tomorrow, 9 o'clock sharp, so we can get out early.